Try hitting it again.
It's a very slow countdown.
Oh, is it still counting down?
Yes.
Oh, that is strange.
It says I have no minutes again. Can you do it,
I'll
Stephen?
do it. Yep.
Okay. Hello, my loves. Welcome to the very first of 5 nights in a row of AP Biology review. We are so happy that you have chosen to spend the next 5 days with us. We will be together from 7 to 10 tonight Eastern time. That's my time. And we're going to go over units 1 to 4. So welcome. Before we start sharing our screen, essentially, I want to make sure you have everything that you need to be successful. So number 1, make sure you have some water, you have some snacks. We are going to take mini breaks throughout the session. Make sure you have something to write with and something to write on. A calculator would also be helpful as well. And we do have a review packet that kind of goes with the session. And there's a green button that you can click on that will take you right to it. And I am Sabrina Shume. I'm an AP biology teacher from Virginia Beach, Virginia. And I've been teaching a really long time, 17 years. And I have 3 amazing TAs with me tonight. And they are going to go ahead and introduce themselves. They're also going to let you know where they are presenting from. And then they're going to let me know if they had to eat the same food for every meal for a week, what meal that's going to be.
Great. I will start off first. So, hi everyone. My name is Madison, and I am one of the student hosts for AP Biology, and I'm coming to you guys from Illinois. One food that I would probably live within like my whole lifetime would be oranges. Very weird food to say, but I like oranges, so that's what I would say.
Hi, I'm Rashi. I'm the other assistant for today. And if I had to eat one food, I think it would just be variations of toast. I feel like toast is such a versatile like food. You can put like jam or peanut butter or just like bananas with peanut butter and toast. So I feel like I'd choose that.
I love it. And we also have another student host, Jeffrey, but I think he's having some technical issues, so he will join us when he's able to. All right, so to kind of get started, essentially you will have one of us presenting with you, and then we also have a chat feature. So if you want to communicate with us in any way or you have questions at any point, please just put it in the chat and we will have somebody helping to kind of check in and then respond to it as well. All right, so let's go ahead and get started. Okay, so we have a really fun time planned for 5 nights in a row. We're going to do half the curriculum today, and then we're going to do the other half tomorrow night, and then we're going to do some practice, and then next Tuesday is going to be a huge grand finale, and then the big day. We got this, you guys, okay? You can totally do this, and we're going to help you along the way. We're going to be using Fiveable. They are the best of the best. Hopefully by now you have checked out their site. You have looked at all the review guides, the videos, the presentations, the study tools, the practice quizzes. They even have an amazing cram sheet like for the morning of that you can use. And as always, please follow us on the social media. Here is the schedule. Here are the 2 different the teacher presenters. It's myself, and then we have another presenter, and we'll be going back and forth. And then these are the different students that will be helping us with the presentations. Here's the overall schedule of what we're going to be talking about. So like I said, tonight is going to be kind of like the nice, easy intro stuff. So chemistry of cells, cellular energetics, communication, cell cycle. And then tomorrow is going to be the big heavy hitters. In terms of percentages of your test, most of your test is going to be on evolution, ecology, and genetics. And so tomorrow is going to be a big, like, heavy hitter night. And then on the night after that, on the 7th, we're going to go over science practices 1, 2, 3, which is going to be like skills and drills, making claims, how you're going to justify answers, making predictions, practice multiple choice and FRQ. And then the next night, we're going to do more multiple choice and practice FRQs. And we're going to focus on more of like the graphing and the mathematical part. So if you are needing a lot of help with the math part, then that's going to be kind of the night where we're going to focus on how to do all the different calculations, like everyone's favorite chi-square and Hardy-Weinberg and rate of change. So fun things like that. And then the huge grand finale after that. So hopefully we'll be able to see you at each of the nights. And now I'm going to pass you over to Madison.
Perfect. So like Ms. Schumann said, we're going to have 5 amazing days with you all regarding the units of AP Biology, as well as doing some practices, skills, and most importantly, the games that us student hosts will be collaborating on. So our agenda today, we already did our welcome and the start of our journey for the Cram sessions. And throughout this meeting, we'll go up units 1 to 4, which is listed here, which is unit 1 is Chemistry of Life, unit 2 being cell structure and function, unit 3 being cellular and genetics, unit 4 cell communication and cell cycles, and lastly we'll end it up with a quick review and just some lasting info regarding our resources and other excuse me, as well as some other information regarding our schedule for this whole these past 5 days. So you might be wondering, how can you make the most out of these Cram sessions? The first thing is definitely take notes. While we want you guys to participate and listen throughout our time, keeping notes will definitely help keep you active and listen actively throughout these sessions. While we'll be going through a lot throughout these 3 hours, but be sure not to write everything down. While we'll be showing you immense amounts of resources, we are not expecting you guys to write all of them down. Speaking of resources, besides our Cram sessions, Fiveable has many more resources that can help provide you for the best studying techniques, as well as information for the AP Biology exam. However, for Cram Hubs 2, we also provide slides, review packets, and replays just for you guys. So be sure to follow along, and if you guys do miss a few Crams, including our past rehearsals, they'll be shared to you immediately after these sessions, so you're able to come up and go through our schedule and just re-watch the videos as many times as you like. Next, our group chat. We do have a group chat, and that is for sharing tips, tricks, and participating when you guys have questions to ask to us student hosts, as well as to Ms. Shumei. And lastly, we do have a Q&A tab, so if you guys have any questions, you can ask within the comments, and they'll be switched over to the Q&A tab on your guys' right side screen. And later on, our student hosts, us, will be able to answer them, and you're more able to actually look at the comments and be able to find an answer for your guys' questions. So do not be afraid to ask along. So before we get started further, we do have some community agreements. First of all, be supportive. This is supposed to be an engaging community for us throughout the 5 days. And the most important thing that we see as a Fiveable community is to be supportive. We all should lift each other up, and this is a way to learn more and be able to grasp more of your learning material throughout the school year. Next, be patient. We do have 5 nights together, and I feel like all of us are very nervous and just very excited to get straight into it. However, sadly, we cannot use only 1 day to get all through the units. So, like Ms. Schumann said, we'll have 2 days going over material, and another 2 days going over practices, and lastly, a grand finale by us student hosts, which will be practices and games for the unit, well, for the course. So we do have 5 nights, and we'll do the best of our abilities to teach you all the things to know for the AP Biology exam. And lastly, be kind. While I did say and discuss that this is an engaging community, it also is a kind community. Us at Fiveable believe that a kind community is one of the best communities to have within these study routes, as a help you guys be more comfortable during our time, as well as be able to maybe make new friends throughout the time being of learning and be able to gauge further into biology. So before we get started, you guys can win $100 by sharing your Cram experience on TikTok or Instagram. So by using the hashtag 5 Crams and tagging at ThinkFiveable in your post and caption, excuse me, you'll be able to possibly win $100. And excuse me, by getting a lot of views, by making us laugh, and we'll select winners throughout the next 2 weeks of the Cram finale, and we'll notify you via DM from our official account. So, without further ado, let's get started. First of all, where are you guys joining from? Let us know in the chat. Ooh, California, we see Texas. Oh, we see a lot of California and Texas. Wow. Chicago. Sadly, I actually don't live by Chicago, sadly, but Ohio, Minnesota. Wow, we're actually like kind of everywhere. That's amazing. Well, still be sure to continue with the chat, but we're going to go along to starting. But before that, we do have a poll, and we're asking you guys, which unit is your favorite between units 1 to 4? So be sure to respond. Looking at the chat, do you see a lot more A's and B's than C's or D's, which is really interesting because I personally favored like cell communication, cell cycles the most. Actually, I'm gonna hand it off really quick to our other student hosts. Which unit between 1 to 4 is your guys' favorite? Okay, not sure if my mic turned off. Whoops. But personally, I feel like hopefully you guys heard me about my favorite, which was cell communication. But I wanted to send it off really quick to our student hosts to see what their favorite unit was between 1 to 4. If you guys like to answer really quick. Well, I believe that our student hosts can write it in the chat, but we can move forward to unit 1. Oh, before we go to unit 1, we're going to lay out really quick the weighting for each unit. So throughout the exam, not all units are going to have the same percentages. You'll be getting some questions that are worth more points as more of a chance of gaining those type of questions for the unit than other units. So unit 1 will be an 8 to 11% chance. Unit 2 will be 10 to 13% chance. Unit 3 will be 12 to 16% chance. And unit 4 will be 10 to 15% chance. So you can see throughout the other 3 units, there will be more shown than unit 1. However, probably the most typical ones you'll see is between units 3 and 4. And also, if you guys do not know, our exam formatting is you'll have 60 multiple choice questions for 90 minutes, which will be 50% of your exam. And for part 2, you'll have 6 FRQs, which is also 90 minutes, and as well as 50% to your exam as well. And here's a little bit more of our exam layout. It's more laid out by our Miss Shumate. She was able to lay this out for you guys. And she does show you the topics throughout the multiple choice as well as the FRQs. And yeah, we can get started to unit 1.
Thank you. And we are going to go through all this. So do not worry. Do not freak out. We're going to do this one night at a time. And you're not alone. Thank you for those that responded to the poll. I know it's kind of, it's still new for all of us, but if you can respond in the chat, and that's totally fine. But also on the right-hand side, you'll see like the little bars, and then there'll be a poll tab that you could kind of click on. And then you'll be able to respond to any of the polls that we are able to put out. And so when I look, I'm surprised that well, I guess I'm not surprised that cells is everyone's favorite. I think it's because it brings you back to maybe like simpler times. And then chemistry of life being the second favorite. That was very interesting to me. All right, we're going to go into Unit 1, chemistry of life. Okay, so these are basic chemistry. This is bonding. This is properties of water. These are macromolecules. Okay, the very first thing we have to go over is going to be the key vocab from Unit 1. And you have this on the review guide that's going to kind of go with the presentation. And again, if you don't have the review guide, it's a little green button at the bottom. Y'all can click on it and the review packet will pop up. And you will also have access to the slides in the Cram hub. So you don't have to write these all down. But instead, I want you to kind of use it as a study tool of you should be able to, on your own, define each of these words just on your own. And you should be able to almost connect them to how they are related to each other. Okay. So now Madison's going to give you a little quiz to see if you can do it. All right, Madison, quiz them up.
Great. For a little quiz for Unit 1, I'll be giving you guys 3 definitions of the terms you guys will be figuring out. So I'll first start off with a definition of one of the terms seen in the slide, and it's up to you guys to figure out which term I'm discussing. So without further ado, I will start with the first definition, and that is a fatty, waxy, or oily compounds that are soluble in organic solvents and insoluble in polar solvents such as water. Nice. Oh my gosh. Across the board. Amazing job, guys. It is lipids. Perfect. So, wow. Great. Oh my gosh. So then next, I'll go to the next definition. So our next definition would be a sugar derived from ribose by replacing a group with a hydroxyl group. So I do see a lot of people saying RNA, which is pretty similar, but it actually is deoxyribose. Not sure why I'm saying that wrong, but it is. I only see 1 or 2 people saying it, but that is understandable. Oh, now I see a lot of people saying great. I feel like definitely like these type of terms are kind of like not as discussed as before. Like DNA and RNA, I believe personally, that was heavily discussed at least in my course in my classroom, but not as much as like the many other like enzymes and types of groups. So we'll go to the last definition I'll be giving you guys, and that is this is covalent bonds that are between 2 amino acids. Great. Yes, you guys are correct. That is peptide bonds. You guys are so prepared. You guys did great, an amazing job. I'll keep coming in with this per unit, but without further ado, I will be giving it now to Miss Umate to go over Unit 1.
All right, my loves, here we go. All right, so Unit 1, like I said, is going to be properties of water, basic chemistry, and then our organic molecules. Okay, so let's go ahead and start with properties of water. So what are some things that we need to know about water? Number 1, we need to know that water is polar. Okay, so when we say water is polar, it means that it's going to have unequal charges, right? So we know that water is H2O, so you're going to have 2 hydrogens and 1 oxygen, and they're going to have different electronegative charge. Oxygen has a huge negative electronegative charge, and then the hydrogens have a positive electronegative charge. So essentially, a water molecule is unique in the fact that it acts like a magnet. So water can stick to water, and water can also stick to other things because of these opposite charges. Does anybody remember what it's called when water sticks to another water molecule? What was that property of water when water sticks to water? Can you guys tell me in the chat? What's it called? I see it. Rochelle's got it. Cohesion. Very good. Cohesion is water sticking to another water molecule. Okay, very good. Now, what is the reason that water can stick to another water? What's the name of the bond that's formed between two water molecules? What is the bond that you'll find between two water molecules? Beautiful, hydrogen bonds, okay? So a hydrogen bond is what's formed between two water molecules. So you need to know that between two water molecules, you have a hydrogen bond. And so essentially what's happening is if you guys look at the diagram that is on the slide, you have the positive end of one hydrogen being attracted to the negative end of another oxygen. And so that is a hydrogen bond. You can think about it as being like just two magnets, two opposite ends of a magnet being attracted to each other. And so the fact that water can stick to itself and the fact that water can stick to other things that are charged, all of that is due to the fact that water is polar. So any FRQ that you guys get about water, make sure you put in the fact that it's due to its polarity, which means that the water molecule has unequal charges. And so when you write your FRQs, you want to make sure that you elaborate on it. So just kind of connect those things together. So any FRQ about water and its special properties talk about the fact that it's polar, which means it has unequal charges, so a positive end and then a negative end. And that's really unique for water in itself. The other thing I want you to know is the difference between a polar molecule and a non-polar molecule. So polar molecules are going to have unequal charges. So we say polar molecules are water-loving because polar molecules can actually break down with water because water is going to have these different charges attract to the other charges and kind of essentially like pull it apart. Molecules that are non-polar are going to have equal charges. So if I have a non-polar molecule mixed in with water, because there's no unequal charge hanging around, it's not going to be able to be broken down. Like oil is considered a non-polar molecule. Something else I want you to know is the difference between hydrophilic and hydrophobic. Okay, so if you see those terms, if something is hydrophilic, that is a polar molecule. It's water-loving. Hydrophobic is going to be non-polar and it's not going to be able to be broken down. Okay, so kind of just remember those. Then the other thing that I always remember is polar bears like water. So polar is hydrophilic. That's kind of my like silly way of remembering that. In terms of other bonds that you're going to find within the water molecule itself, between the oxygen and the hydrogens are going to be very strong covalent bonds. Those are strongly bonded. But like I said before, between water molecules are those hydrogen bonds. Okay, so make sure you know the type of bonds that are there. Here are some unique properties of water. Again, always due to the polarity. So always connect all the properties of water due to the fact that it's polar, okay? So when we look at the special properties, we talked about cohesion and adhesion. Adhesion is when you have water sticking to other charged molecules. And by sticking, I just mean it's those hydrogen bonds. Ice is less dense as a solid. So the unique property about water is that as you lower the temperature of water, the water molecules kind of spread out and they form these big spaces called lattice structures. And so they kind of just get really spaced out and they kind of shake a little bit because it's really, really cold. And those big lattice structures, those big spaces allows the water to be less dense and actually kind of like float, which I think is kind of interesting. And that's why like in the winter, you're going to have like, you know, the lake's frozen over, but it's not frozen all the way down. And that's why the animals don't die, thank goodness. Has high specific heat. So water takes a lot of energy to heat up and then it holds on to that energy. It has a high heat capacity for like a really long time. So I live in Virginia Beach, so I'm right near the ocean front. And essentially, if I were to go to the water during the day, the water is going to be a little cold. But if I go back at night, the water is actually warmer, which is unique. And it's unique because essentially it had all day to heat up and then it holds on to that water. And so that's what we mean by it has a high specific heat. The other property of water is going to be that it's a universal solvent, right? For those molecules that are polar, it's going to go ahead and be able to attach and then kind of break it down. Okay, so my question for you is, I have a 6-year-old and I have a 9-year-old, and one of their favorite things to do in the summer is to blow bubbles. So when we're blowing bubbles, what property of water is being shown? What do you guys think? What properties of water allow for my kids to be able to blow bubbles? Great, it's both of them. Cohesion, very nice, and also surface tension, right? It holds it in. All the water molecules are sticking together, and then eventually it's gonna pop. Very good, perfect. You guys are rock stars. Okay, before we move into the hard stuff, let's take a little break. So here's another poll. Okay, and Rosh is gonna help us present it.
Yeah, so let's do a poll on what is your favorite type of vacation? A beach vacation, a ski trip, or a staycation where you stay at home. And so if you click on the right side on the polls option, you guys should be able to see the poll now and vote. Wow, okay, so it definitely seems like most people are fans of the beach. I am from Florida, so 100% support that. I think that's the thing I am most looking forward to this summer is going to the beach. Yeah, over 50% of people say beach. So I am on your side, beach lovers. Okay, and let's do a few concept checks just to make sure we understand everything that we've covered so far. So the first question is, what property of water is responsible for water moving from roots to shoots? So A, surface tension, B, high specific heat, C, cohesion slash adhesion, and D, it's more dense as a liquid. So I'll give you guys a minute to drop your answers into the chats. So I am seeing a lot of C, and that is in fact correct. The cohesion slash adhesion is water being able to stick to itself, and that's what helps it move from the roots to the shoots within a plant. And then surface tension, remember, is like making it more difficult for water molecules to split. And so that's like how an insect lands on the surface of a lake and it's still able to crawl. So that isn't quite the property of water that's being used here.
All right, perfect. Good job, you guys. Thank you, Rashi. All right, so properties of water was probably the easier of the chemistry unit. So now we're going to kind of go into the harder things. Okay, so basic chemistry, we all know that in terms of molecules themselves, we know that molecules are made up of atoms, right? And atoms are made up of protons, neutrons, and electrons. We know that elements are going to be atoms of all the same amount of protons. And so what we really focus this unit on is studying organic molecules. So all organic molecules are going to contain what element? What element do all the organic molecules contain? Tessa's got it. Very good. Carbon. Okay. So we're going to be looking at molecules that contain carbon that make up us, and we call those organic molecules. And so the unique thing about carbon, and it has a very special property. Does anybody remember the special property of carbon? Like, why is carbon special? And I'll give you a hint. We say it has tetravalence. What does that mean? Does anybody know what tetravalence means? What's unique about carbon? Beautiful. Yes, it can form 4 bonds. Beautiful. Great job, you guys. Okay. So carbon, it makes up all organic molecules. It's unique in the fact that it can form 4 strong bonds with other elements. And we call that ability tetravalence. Now, carbon can form all different types of structures. And so what I want you to really know is what an isomer is, and then essentially the different types of isomers. Okay, so there are 3 types of isomers. So an isomer basically means you have 2 molecules that are going to have the same molecular formula, but they're going to kind of be put together differently. And one of the main themes in AP biology I want you to remember is that structure determines function. And that's with almost every single unit that we study. So the moment you change the structure, you're going to go ahead and change the function. So structure equals function, and it's very important. So in terms of the three different types of isomers that you need to know about, there's structural isomers, geometric isomers, and then enantiomers. So a structural isomer means if I were to look at the molecular formulas, the structures are just different. That's it. Okay. So one might be linear, one's going to be maybe more circular, one's going to look like the letter T, one like the structure is just different, but they have the same amount of elements and the same number of elements. Okay. Geometric is going to be when you have a double bond and one molecule is going to look like it's rotated around a double bond. So anytime you see a double bond, then you know it's a geometric isomer. Okay, same molecular structure as well, or the same formula, but it's going to be rotation around a double bond. In terms of enantiomers, those are going to be mirror images. So for example, if you look at your hands and you put them together, they're going to look the same, right? But if I open them up, on one hand, I have my thumb on this side, and one hand, I have my thumb on the other. They're mirror images, but they're not the same. So those are called enantiomers, okay? So isomers are going to be molecules that have the same structure, same atoms, and then there's three different types. We have structural, so if I look between two molecules, the structures are just different. They just have different shapes. Geometric is going to be around a double bond. That's the only one around a double bond. And enantiomers are going to be molecules that look like mirror images. All right, so if we go into what are called macromolecules are large organic molecules. There's some terminology you need to know. You need to know the difference between a monomer and a polymer. Between those two words, which one means a single unit? Is it monomer or polymer? Which one means a single unit? Does anybody remember? Monomer, very good. Monomer means one, right? Poly means many. So when we talk about organic molecules, I want you to know for each of them the monomer. Okay, you should be able to tell me the monomer of a carb, a lipid, a protein, and a nucleic acid. When we start putting several subunits together, that's going to be called a polymer. So in order to understand our macromolecules, you need to know how we actually form them and then also how we break them down. There are two processes that you need to know: dehydration synthesis and then hydrolysis. Okay, so you should know the difference between these two. Between those two, which process do we go through when we want to break down a molecule? Which one's going to be used to break down a molecule? Is it dehydration synthesis? You can put DS or H for hydrolysis. Beautiful, hydrolysis. Hydrolysis is what the process we use to break down molecules, okay? So when I take in milk and I have the sugar in milk, which is a disaccharide, does anybody remember the name of the sugar in milk? What's the name of the sugar in milk? It's a disaccharide. Lactose. Beautiful, okay? So when I drink milk and I need to break it down, I essentially am going to add water to it, okay? And it's going to be mediated by an enzyme. So an enzyme is going to add water in between this huge disaccharide, and it basically breaks apart the molecule, forming those two simple sugars, okay? So we go from something big to something small. And so hydrolysis, the way I think of hydrolysis is I think about like digestion, right? So when you're digesting something, you're going to eat the big food, you're going to break it down into your stomach. And that's why your body is always craving like water, because it needs water in order to break down the food, to go through hydrolysis to break it down. Now, if we need to build something, like we need to build a protein, we have to put together some amino acids. We essentially are going to remove a hydroxyl group from one part and then a hydrogen from another, and then it kind of creates a sticky end, and it kind of builds together, okay? So when you remove water, when I think about like when I'm really like dehydrated, the water has been removed, that's how I build molecules, okay? So dehydration synthesis is going to help us with building molecules. Hydrolysis is going to be the addition of water to break molecules down. So when we talk about going from monomers to polymers, then we're going to be building something up. So that's going to be through dehydration synthesis. And we're going to use that for the entire chemistry unit. So make sure you understand the differences between the two. All right, Rashi's now going to go ahead and do our second concept check and see if you can understand another process.
Okay, So 2 amino acids are brought together during protein synthesis at the ribosome. What process forms the peptide bond between them? A, dehydration synthesis, and B, hydrolysis. So I'll give you guys a minute and drop your answers into the chat. So I am seeing a lot of A, or dehydration synthesis, and that's correct. Dehydration synthesis is building up molecules and they require energy. And remember, hydrolysis breaks down the molecules and releases energy.
Great job, you guys. So far, we're doing great. All right, so let's go ahead and go into the macromolecules. So what are the things you need to know? You need to know the monomers of them, some basic characteristics, kind of what elements make them up. And so you have this nice little graphic organizer to kind of help you out. And then we also have one at the end that so you don't have to copy everything down. So there is so much to cover in Unit 1. I'm going to just try to hit the big hitters and just kind of do my best, okay? So let's go ahead and go through like the big ideas of what you need to know for each of the macromolecules. So we'll start with carbs, okay? So carbs are made up of carbon, hydrogen, and oxygen. And what's unique about this macromolecule is that you are going to have 2 hydrogens for every oxygen that you have. So it's kind of called an N-to-N relationship, and that's how you recognize that it's with the carb, right? So your carbons and oxygens are always going to be found in equal amounts, and then your hydrogen is going to be double that, okay? So if I gave you a molecular formula of C6H12O6, if it has that N-to-N pattern, you automatically know it's a carb, okay? So I want you to kind of remember that. So carbs contain CHO, carbon, hydrogen, and oxygen, and it will have the N-to-N pattern always. Okay, so again, if I showed you one and it said C4H8O4, you'd go, oh, that's a carb. I can tell right away. And carbs can be linear. It can be in the circular format right here. We're always going to have that particular format. We're going to use carbs for short-term energy and also for structural support. One sugar is called a monosaccharide. Saccharide means sugar. So carbs is just sugar molecules or saccharides. When you have one sugar molecule, it's called a monosaccharide. When you have 2, that's going to be a disaccharide. And then when you have more than 2, you know that we're going to have polysaccharides. In terms of the examples that you need to know, we know that sugars are going to usually end in OSE, like glucose, right? That's going to end in OSE. Lactose is going to end in OSE. You need to know that the disaccharides would be the ones with 2 sugars put together. And then some polysaccharides examples that I want you to know are going to be some plant examples and animal examples. So I want to focus on the polysaccharides, okay? So in terms of the plant examples, the difference between starch and cellulose is something that you kind of do have to be able to understand, okay? So starch has a unique structure where it's going to have its hydroxyl group kind of located on the bottom. You can kind of see the picture in the lower left corner. And so we're going to call that the alpha structure. And when you have a whole bunch of those in a line, and that would be a polysaccharide, and you digest, you're going to eat the starch, and your body is going to be able to break it down, and then we're going to get some energy that way. Okay, so you're going to find starch in things like potatoes. So I can eat a potato, and my body can break it down. Now, what happens is we have something called cellulose. Cellulose is something that we can't break down. So cellulose is going to be a very hard, it looks like almost like a fiber. And that the point of that is for structural support. Okay, so we had the starch and we got some energy from it. But in terms of the point of cellulose, it's more for structural support. When I think of cellulose, I think of a celery stalk. And if you were to like break a celery stalk, those fibers that you see, that is cellulose. And your body is not able to break that down, because what happens is we're going to have our monosaccharide that's going to have this alpha form right here with the hydroxyl on the bottom. But then it connects to another monosaccharide where the hydroxyl is on the top. And it alternates. It goes up, down, up, down, up, down. And so when the hydroxyl is at the top, that's called the beta form of the monosaccharide. And because the structure changes over and over and over, we don't have any enzymes to break that down. So we can't break down the cellulose. So instead it turns into fiber. So it is important for you, but we just can't break it down and get any energy from it. It just becomes fiber and like cleans out your digestive system. In terms of the animal examples, we're going to store energy in the form of glycogen. It's a super branchy molecule that we have in our liver. We are going to also have a structural example called chitin. And so chitin is going to be like if you had like a crab, that hard outer covering, or if unfortunately you've seen a termite in your life and you like squashed it and it was like crunch, that is chitin. It's a polysaccharide. Okay, so we use that for structural support. So carbs are short-term energy. We can have one sugar monosaccharide, disaccharide, polysaccharide, and it's going to usually end in OSE and then contain the N2N pattern. Okay, so that's the big picture of carbs. All right, so the next one, lipids. What I want you to know about lipids, I want you to know that all lipids are nonpolar. All of them. That's a unique property for lipids, is that they're going to be nonpolar. They are going to contain the same elements as carbs, C, H, and O. The difference is we're going to have a lot more hydrogens. Okay. Does anybody remember the name of the tails found in fatty acids? Like, what do we call something that has a bunch of hydrogens and carbons? It contains a lot of energy. Does anybody remember what that's called? What contains a lot of hydrogens and carbons? It's not a trick question. Ooh, maybe it is a trick question. Oh, I saw it. I saw it. It's a hydrocarbon. I didn't mean to be tricky. Hydrocarbon, okay? So when we look at these really, really long tails found in fatty acids and lipids in general, they're going to be long chains of hydrogen and carbons, and those are hydrocarbons. And hydrocarbons contain lots and lots and lots of energy, okay? So lipids provides us lots and lots and lots of chemical energy. It also is going to give us some sort of structural support, right? The other unique property about lipids is that when we had our sugars, they kind of link together, but lipids instead are going to almost like stack on top of each other. And so the other unique property of lipids is that they can't link together. They more of stack on top of each other, okay? Now, we can also form a water barrier, right? If we look at this very special lipid called a phospholipid, we're going to have a phosphate at the top, which is the head, and then we're going to have these fatty acid tails, okay? So this top part is going to be hydrophilic, so that part is going to be charged, so it's going to like water. And then the tails are hydrophobic because that part's actually made of lipids, okay? So essentially, when we put a bunch of these phospholipids together, we get the phospholipid bilayer. So you're going to have a nice little water barrier. Another example of our lipids is going to be like waxes. Waxes kind of have this funky little formula shape right here. Another example is going to be like steroids, cholesterol, sex hormones. They kind of have these 4 fused rings. It's called an isoprotein. These are all lipids. So I want you to know that lipids are going to be nonpolar, okay? They're not going to form like monosaccharide, disaccharide, polysaccharide. They kind of stack on each other. And then the last thing you need to know about lipids is that there are going to be 2 different types of lipids you need to know: saturated fat and unsaturated fat. So make sure you know the difference between the 2. So my question for you guys, saturated fat, unsaturated fat, which one contains all single bonds? Which one has all single bonds? Is it saturated or unsaturated? Beautiful. Okay. The way to remember that is the letter S. Saturated fat is going to be solid at room temperature and contains single bonds. Very good. So SSS, saturated fats are solid at room temperature and they contain all single bonds. Great job, you guys. Unsaturated fats are going to have a double bond. And when you put in a double bond, it's going to bend the molecule. Okay. So when I have 2 unsaturated fats trying to stack on top of each other, they start sliding off each other. So it can't form a solid. So your unsaturated fats are going to be your liquids at room temperature. Okay. So saturated fats are going to be all solid room temperatures, single bonds. Unsaturated fats are liquid at room temperatures and they're going to go ahead and they're going to contain a double bond. All right, proteins. What do we need to know about proteins? We need to know that proteins are made up of amino acids. We need to know that they are the most diverse group that we have. We need to know the basic structure of the amino acid. So I have the structure in the lower right corner. And I don't know if you guys ever put together, but why we call it amino acid is all amino acids have a central carbon. They have an amino on one side, and they have a carboxyl acid on the other. So it's called an amino acid. The only thing different between all the amino acids is going to be the R group. Okay, so R group is going to be the one thing that distinguishes the difference between all of our amino acids. So some R groups are going to be polar, some are going to be charged, some are going to form disulfide bridges if it contains sulfohydryl. And so this is kind of where those functional groups are going to come into play. But for the most part, I need you to know that proteins are made up of amino acids. I need you to know that it's the most diverse group we have. And what is the most famous protein that we study in AP Bio? Does anybody know what's that protein that we study? It's very specific. We talk about it a million times. What protein did you learn a lot about in AP Bio this year? Yes, enzymes, 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 enzymes. It's all about the enzymes, and we will talk about the enzymes, okay? So I need you to know that enzymes are a type of protein. Now, what else do we need to know about enzymes or about proteins in general? You need to know how proteins form and essentially the 4 different steps that we have, okay? So when we have a protein being created, does anybody remember the name of the organelle that helps us make proteins? What is the organelle that makes proteins? Some of them are attached, some of them are free floating. What? Very good. Great job, you guys. Ribosomes. Okay, ribosomes help make proteins. So when we are on a ribosome and we are just putting together all those amino acids, we are going to have what's called the primary structure. So as that particular protein is being made on the ribosome, that is called the primary structure, which is step 1. Think primary, step 1. Okay. Between those amino acids are called peptide bonds. And when we go ahead and link them all together, that's called a polypeptide. So once we have our linear sequence created, it's now going to do its first folding, and that's called secondary structure or the second step. So each of the steps we call primary structure, secondary structure, tertiary, and quaternary. I just remember step 1, 2, 3, and 4. Once we have local folding happen, there are 2 different forms that you can have. You can have it kind of curl up, kind of like a little piggy tail, right? Or like Tigger's tail, and it's really bouncy. If our final protein has to be super flexible, then we're going to form this alpha helix structure. If our final protein has to be very strong, then we're going to form what's called the beta pleated sheet. So the end structure determines the path it's going to take. So again, if it's going to be flexible, we're going to go from primary to secondary and form the alpha helix. It's just going to coil in on itself. Or it kind of starts to kind of condense and stack in on itself because it has to be strong at the end. Tertiary structure is when you have the first time all the R groups start interacting with one another. And now we're starting to get a structure start to form. So all the different charges are coming together. The disulfide bridges are starting to form. Anything that's hydrophobic moves to the inside. Anything that's hydrophilic moves to the outside. So this is the first time all of those are going to kind of form together. And then quaternary is going to be when you're going to have all the different polypeptide chains come together. All right, so now let's go ahead and have Rashi do a concept check with you guys.
In a folded protein, where would hydrophobic R groups most likely be found? A, on the inside, or B, on the outside? So I'm seeing a few mixed answers, but the answer is A, on the inside. Amino acids with nonpolar hydrophobic R groups like to cluster inside of the protein and leave the hydrophilic amino acids on the outside so they can interact with the surrounding water molecules. And the hydrophobic R groups on the inside don't have to deal with the water molecules. And one more concept check. In certain proteins, disulfide bridges form between the cysteines of two amino acids. In what level of protein folding will this occur? A, primary, B, secondary, C, tertiary, or D, quaternary? So drop your answers into the chat. I'm seeing a lot of C, tertiary, and that is correct. So the primary folding is just a sequence of amino acids in a polypeptide chain. Then the secondary level of folding are those folded structures within a polypeptide because of interactions between the atoms. And the tertiary level is the overall 3D structure of a polypeptide. And this is where bonding occurs between different atoms, including disulfide bridges. And now we're going to do one mini break. So I'm about to publish. You guys should be able to see a poll on the right side. What is your favorite macromolecule dish? The first answer choice being pasta and bread, the second one being steak and potatoes, or dessert. Me personally, I am a pasta and bread girl. I did talk about eating toast for the rest of my life earlier, so I guess that's on track. I'm also vegetarian, so steak and potatoes is kind of automatically out, but I really love pasta. I made this great tomato pasta the other day. It was really good. So I am seeing a pretty much equal split between steak and potatoes and dessert, but pasta and bread is in the lead with over 50%. So I support you guys and fully agree.
All right. Thank you so much, Rashi. So I saw you guys had some questions about disulfide bridges. Don't fret, no big deal. So one of the R groups contains a sulfohydryl group, which is a sulfur attached to a hydrogen. And what's unique about sulfohydryl is that sulfohydryls want to hunt out other sulfohydryls. And when they do, they like are best friends and they like link up together and they're like, oh my gosh, and they form this little bridge between them. So the best example would be people that have like curly hair. I don't have curly hair. I totally wish that I did. But essentially what happens is in the structure of the hair, they have a bunch of sulfohydryl groups that find each other and it keeps the hair nice and curly. And so what happens is sometimes people with curly hair, they want to go ahead and they use heat and they use a straightener to straighten it out. And what they're doing is that heat is breaking the bond that's formed between those two sulfohydryl groups. And it breaks, and then the hair is going to go ahead and go straight. But with time, those little sulfohydryls are then going to find each other again. Oh, my curly hair community. I love it. I wish I had curly hair. So sulfohydryls is nothing more than a special R group that contains sulfur that's going to hunt out essentially another sulfohydryl group and then form that disulfide bridge. And that's all that that's going to be. All right. So the very last macromolecule I want to talk about are going to be nucleic acids. The only thing I want you to know about nucleic acids is that they're made up of nucleotides and that a nucleotide contains a phosphate, a sugar, and a base, okay? The only three examples you have to know are that DNA, RNA, and ATP are going to be your three examples of nucleic acids. That's it. DNA, RNA, and ATP. Okay, we know DNA, RNA, and ATP are the three examples. They all contain nucleotides. So my question for you is if you had to guess, how many phosphates group are found in an ATP molecule? How many phosphates are found in an ATP molecule? Adenosine triphosphate. How many phosphates are there? Very good. 3. If I were to remove a phosphate from ATP, what does it become? Just going to ask you. What does ATP become if you remove a phosphate? There it is, ADP. Very good. Okay, so we know ATP is going to be our energy currency essentially. And when we lose that third phosphate, we're going to release that energy from that third bond. Then we go to ADP and we kind of go back and forth because it's a coupled reaction. Very good. Okay, so if I were to look closer at a nucleotide, which is a phosphate sugar in a base, okay, I want you to know that when I look at those nitrogen bases, there's actually two different categories of nitrogen bases. We have what are called purines and we have what are called pyrimidines. And so in terms of the different bases that we have, we usually refer to them as like A, T, C, G, right? Adenine, thymine, cytosine, and guanine. Now in RNA, does anybody remember the base that we use in RNA? We don't use thymine. What do we use in RNA? What's it replaced with? There it is, uracil. Okay, so I always remember like RNA loves you, and it's just kind of like a cute thing that I say to my kids. And so in RNA, we're going to use uracil, but if it's going to be DNA, there's going to be that thymine. Okay, so you need to know the difference between a purine and then essentially our pyrimidines. Okay, so my thing that I always remember is I remember the saying pure as gold. Your purines are A and G, and everything else is going to be your pyrimidine. Okay, so remember, pure as gold. Your purines are going to be A and G, and everything else is a pyrimidine. And then the other unique thing about the two types of nitrogen bases is that they're going to be connected with a purine and pyrimidine. So A always forms with T, and C always forms with G if it's like a DNA molecule. Okay, if it's RNA, then it would be A and U. What you need to know are the different types of bonds or how many hydrogen bonds are found between these nitrogen bases. So I put this little thing on here at the bottom right. I just want to explain it. So I have my students remember A2T C3G, A2T C3G, A2T C3G. It kind of gets catchy, right? And so what happens is by remembering that, that's going to help you remember that A pairs with T, and there are two hydrogen bonds found between them. And C pairs with G, and there are three hydrogen bonds between them. Hydrogen bonds are easily broken. We know that we're going to break it for DNA replication, but you need to kind of remember that little saying. So A2T, C3G, two hydrogen bonds between A and T, and then three between C and G. Very good. Another way of remembering, somebody just put that, there's going to be apples in the trees and cars in the garage, right? So that's something else that's going to help you kind of remember those different sayings. All right, so when I look at a DNA molecule, I want you to know that the strands are called anti-parallel. Here's kind of like a drawing that someone has created, and you can kind of see how on the left-hand side, you kind of have all of those nucleic acids kind of like stacked on top of one another. And on the right, they're kind of upside down because they're anti-parallel, kind of going in opposite directions. And again, you already know the type of bonds formed between them, which is going to be hydrogen bonds. And so we know that what we have to do is we're going to have to go ahead and break those hydrogen bonds in order to replicate. And so the other teacher is going to go over that on the second night. All right, so let's go ahead. And with Rashi, she's going to go ahead and do another concept check.
Using Chargaff's rules, find the percentage of thymine, guanine, and adenine within the given DNA molecule when given the percent of cytosine is 14%. So A has 36% thymine, 40% guanine. Actually, I will not read these answer choices out loud, but I'll let you guys take a few minutes to do some quick math over here. So I am seeing a mix of A and C. The answer is A. So we know that if cytosine is 14%, that means that guanine also has to be 14% because cytosine and guanine are like partners. And that leaves 72% for adenosine and thymine. But remember that adenosine and thymine are splitting that 72% within themselves. So adenine and thymine are splitting that 72% within themselves. So adenine will get half of that, or 36%, and thymine will get the other half, which is the other 36%. So the answer is A. And make sure you guys know how to do some quick math like this, because you might be asked to do like some mental math or some like fairly simple math on the exam as well.
Perfect. Thank you, Rashi. Beautiful. All right. So in terms of macromolecules, big pictures, what do we need to know? The monomers. Carbs are made up of monosaccharides. Lipids are going to be your fat. So anything dealing with fatty acids, and it literally will have the word fat in it. We know that's going to be a lipid. We know nucleic acids are made of nucleotides. The words look the same. And proteins are made up of amino acids. Big picture things that I want you to know is going to be carbs are going to be short-term energy. We know that lipids are going to be all nonpolar. Nucleic acids, the 3 examples we had to know were DNA, RNA, and ATP. And then proteins are the most diverse group that we have. And then enzymes are a type of protein. Okay, we're going to try something. So I need you to kind of bear with us as we figure this out. But there's going to be a poll feature. So I need you to get your cell phone or iPad, anything that can read a QR code. Okay, so go ahead and take a second, go find something that can scan a QR code. And we're going to go ahead and we're going to launch it's kind of be like a Kahoot where you're going to get a particular code and then you're going to go ahead and try to answer some questions. Okay. All right. Bear with me. All right, if you could go ahead and scan that QR code for me. We're going to do some practice questions. I like the little peppers. Those are bringing me joy. Keep them coming, you guys. We got this. They're making me laugh. All right, about 30 more seconds. We're going to go ahead and hit start. All right, come on. You got this. About 10 more seconds. Let's see how we do on Unit 1. All right, my loves, here we go. About 10, 9, 8, 7, 6, 5, 4, 3, 2, and possibly 1. All right, we're going to go ahead and start the quiz. Okay, so let's see how we do. So it should show up on your own device, and then you can select the answer. All right, here we go. Question 1. I'm going to let you read it, and then you guys go ahead and select it. All right, let's see how we did. All right, beautiful cohesion, adhesion. Great job, you guys. All right, let's try the next question. The level of protein structure that allows multiple proteins to come together. So more than one polypeptide sequence. Which structure was that? Was that 1, 2, 3, or 4? Beautiful. 4. Okay, so 1 was just a linear sequence. 2 is going to be when it forms the alpha helix or beta plated sheet. Tertiary is when the R groups come together, and quaternary is the last step where more than one polypeptide sequence is going to come together. All right, let's try another question. If you were to remember how water diffuses, how does it diffuse? Very good. High to low. Okay, water's always going to diffuse from high concentration to low concentration. Very good. All right, let's see. Which statement best describes why water is a polar molecule? We talked about this special property of polarity. See if you can figure out which one describes why water is polar. Very good. Good job, you guys. All right, so let's go ahead and we're going to move on to our next unit. Okay, so let me go ahead and share my screen. Good job, you guys. Take a little break if you need it. Have a nice little stretch, grab some water. We're going to go right into Unit 2. Have a stretch while you can, you guys. All right, you guys, I'm having some technical difficulty. Give me 1 second. Unit 2 is going to be about cell structure and function. Luckily, it's going to be one of our easier units. So Madison, if you could come in and let's see if you can give them 3 definitions and let's see if they can figure it out inside the chat while I try to figure out what's going on.
Of course. First of all, congratulations on finishing Unit 1. I am so proud of you all. So I will give out 3 definitions, and you guys have to guess, once again, the terms. So the first one, just a little bit simple, is what is the term for basically when a cell dies, when there's multiple steps that lead to the death of a cell? Great. Yes, you guys are correct. It is apoptosis. I personally think it's not talked about, like the actual term, personally. I feel like a lot of students don't know. For my class, we barely knew it until a few weeks ago, so that's why I decided to say it here. But great. Next one. So I'm going to talk about a specific organelle this time, since this unit does heavily discuss on organelles. So what is a membrane enclosed organelle that contains an array of enzymes that are capable of breaking down biological polymers? Perfect. Lysosomes, you guys are correct. Within this unit, there's a lot of functions you have to remember on organelles, and I feel like lysosome is one that's not as discussed about, rather than the mitochondria, which I feel like everyone at least has an idea of what that organelle could do. And lastly, I'm going to ask you one more definition. So, what is a kind of protein that is found embedded within the plasma membrane of the cell? And I'll give you a hint. It's involved within the diffusion of ions and water molecules. So I see a mix of integral and channel proteins, but the actual answer is channel proteins, because channel proteins are involved in actually being involved in diffusing those ions and water molecules through the plasma membrane due to the hydrophilic environment between outside the plasma membrane and inside the plasma membrane. Great. So not see Miss Shoemate. I see that she's still currently working on technical difficulties. So, oh, perfect. Great. Okay. Please stand by, everyone, for 1 second.
Hi, okay, I'm going to try and share my screen. I don't have much experience with this, so please bear with me as we go through the rest of these. So this is an overview of Unit 2. And so Unit 2 has a bunch of different parts on just about cells. So we're going to talk about cell structure and function today. And so first, let's start off with some of the major characteristics of life. As we all know, I mean, life is all about reproduction and energy, adaptation, cells, etc. But the main focus that we are going to discuss in this unit is cells and how these itty bitty cells go all the way into making up an organism. So it's very important to make sure you guys are following along. So let's do a quick organelle review. Concept check number 1. What do ribosomes, mitochondria, and chloroplasts all have in common? A, they contain nucleic acids. B, they are found in prokaryotic cells. C, they are found in animal cells. Or D, they are found in all living things. Okay, I'm seeing a variety of answers. So in this case, it's that they all contain nucleic acids. Remember that chloroplasts are found only in prokaryotic cells in plants. So that kind of rules out C and D immediately. And then concept check number 2. According to the endosymbiotic theory, A, DNA and nuclei of eukaryotic cells exhibit a symbiotic relationship. B, chloroplasts and mitochondria were once free living prokaryotic cells. C, membrane-bound organelles evolved from single cellular prokarya that were enveloped by ancestral eukaryotic cells. Or D, both B and C. So this one is fairly longer, so I'll give you guys a few minutes.
Right, good job, you guys. Thank you, Rashi. All right, Rashi, can you go ahead and show them the right answer? Very good. You guys nailed it. It was D. Good job of B and C. So if we could then go to the next slide, we're going to actually talk about the endosymbiotic theory. Hopefully you guys remember this theory. It's this idea that kind of explains the evolution of complex organisms, how we kind of go from something that was very simplistic to something that's a lot more complex. And so the theory is going to state that you kind of had this prokaryotic cell, and he was kind of like a big eater, right? He just kind of went around eating big things. And he kind of came across these two independent prokaryotic cells, and they were both able to transform energy in some way. And so one of them we know to be mitochondria, and the other one is known to be a chloroplast. And so when this big eater came and enveloped these particular mitochondria and chloroplasts, it was able to transform energy. And so instead of breaking it down, essentially this one bacteria guy was like, oh, well, if we can just all kind of work together, then let's just work together. And so that's kind of the idea of the endosymbiotic theory. Now, there are several pieces of evidence that support this theory. Does anybody remember what is unique about chloroplasts and mitochondria that we use as evidence to support the endosymbiotic theory? There's lots and lots of pieces, but what is unique about mitochondria and chloroplasts that gives us the support of the endosymbiotic theory? What's one of them? Great job, you guys. Perfect. You're saying all the right answers. They both contain their own DNA. They both are going to be able to transform energy. They both have double membranes. Perfect. Okay, they can replicate independently of each other. They can move independent of the cell itself. Beautiful. You got it. Great job, you guys. So if we could go ahead and now talk about cell size. Cell size is something that's very important, right? It is important to know that cells want to have a high surface area to volume ratio. They want to be able to interact with the environment very quickly, transform energy, but they can't be too big, right? They can't be too big because it would take too long for things to go in and out of the cell, but it can't be too small because it has to hold all that stuff inside of it. And so cell size is very important to the actual cell. So in the next slide, we can talk about essentially diffusion and cell size. So here's an experiment that a lot of us did in our classes. You might have done different variations of it, but we have 3 different size cubes, and you're going to put it into some sort of solution and watch the rate of diffusion. And so if we look at diffusion, which is just the movement from high concentration to low concentration, we can recognize that the smaller cell has almost all that pink gone. So whatever we put this in, it diffused to the inside of it. So I want you to remember that cells want to have a high surface area to volume ratio. So if you think about something like a neuron, right? A neuron, where do we find neurons? Do you guys know where we find neurons? This is not on the test, but I just want to know if you guys know where do we find neurons? Where do we find neurons? In the brain. Very good. My AP Psych people should know that one, right? You guys just had your test. So neurons are highly branched cells in terms of their structure. And so the cells are going to have lots and lots of branches to increase the surface area without actually increasing the volume itself. Okay. So I would write down in my notes, I put a little star that cells want to have this high surface area to volume ratio. So on the next slide, what you'll see is that you're going to have 4 different sizes of cells, and they all have different surface areas and volumes. And you might be asked to calculate the surface area and volume ratio. You're going to be given a formula sheet, so make sure you bring a calculator on the day of the test. Make sure it's approved by College Board. If you go to College Board, it's going to go ahead and have the approved calculators on there. And every formula you need is on that formula sheet, okay? And so what you'll do is you're going to find the surface area of a cube, the volume of a cube, and all you have to do to figure out the ratio is legit just divide it. So surface area to volume ratio is just a division. So on the next slide, you'll see that we have a chart set up, and it's going to have the surface areas, the volumes, and you just divide them. That's all that you do, and you're going to come up with a ratio, okay? So just remember that they want to have the higher surface area to volume ratio. That makes that the cell is going to be more efficient. All right, now we have a concept check coming up. Okay, so on the next slide, let's talk about the concept check. Lymphocytes are specialized white blood cells. They help provide specific immunity against pathogens as part of the adaptive immune system. B lymphocytes or B cells produce antibodies, which is a specific type of protein. Based on the information, which organelle will be high in the number in a B lymphocyte. Even though you don't know what any of these things mean, the word that I'm kind of cluing in on is that it's a protein. So which one of these organelles are you going to find a lot of if we know that we have to make a protein? Very good. All right, the answer on the next slide is going to be C, ribosomes, right? Ribosomes are the site for protein synthesis. So the AP exam is going to do a lot of these things where it's going to throw like really big words at you. Like just, it's okay. Take a deep breath. It doesn't matter. You didn't know what any of those were. We all know that proteins are made up of amino acids, and we know that proteins are made up of ribosomes. Very good. All right, so on the next slide, we have the specialized structures and functions. We should know some similarities and differences, right? Like what cell contains chloroplast? Is it a plant cell or an animal cell? What contains chloroplast, you guys? Plant cell or animal cell? Plant, beautiful, very good, okay? So those are not gonna be too difficult for you guys, which is beautiful, all right? So we're gonna go ahead and move into the harder part of cells, which is gonna be the cell membrane, okay? So if we go on, look on the next slide, we know that some molecules can easily diffuse and some molecules cannot easily diffuse because of this phospholipid bilayer, okay? So these phosphates heads are going to be charged, so they're hydrophilic, And the tails are hydrophobic, and it creates this water barrier between the inside and the outside of a cell. So things that are hydrophobic can easily go through the hydrophobic tails because like, right? So essentially they're like, oh, come on in. But something that is going to be too large or charged, it can't diffuse through. So we have to talk about the differences between active and passive transport. Okay, so let's look at a concept check. All right, so looking at what we have here, determine the polarity of the signaling molecule and then defend your choice with evidence from the diagram. So you're going to be given tons and tons of diagrams like this, okay? I just want you to look at what they gave you. Take a deep breath. Don't freak out. Most everything you need is going to be in the diagram. So let's dissect what the diagram gave us, okay? So here's the cell membrane. We have the cytoplasm out here. Here's where the nucleus is. We have a signaling molecule come in, and it's able to go through this membrane and go ahead and process transcription. All right. Good job, you guys. All right, let's look at the next slide, and it basically says exactly what you guys said. It is nonpolar because it can go right through that. Beautiful. You guys nailed it. Great job. Good job, you guys. All right, now let's talk about the different types of transport that we have. We have active transport and we have passive transport. Between active and passive, which one requires energy? Is it active or passive? Which one required energy? Active, beautiful. Active requires energy, passive does not. Okay. Things in nature like to go from areas of high concentration to low concentration. And in this particular part, I like to do like a little word exchanging. And if you get confused between high and low and concentrations, if you replace it with the word elevation, it's kind of like a little study tip, right? So if I were to ask you to go from a high elevation to a low elevation, you could like easily just roll down a mountain. No big deal. Like I can easily, just gravity will push me down, okay? So things want to go from high to low. So they want to go from high concentration to low concentration. That's what they want to do. If I then asked you to go from the lower elevation and I asked you to push a rock to the very top part of that elevation, that's going to require a lot of energy. So active transport is going from a low concentration to a high concentration, kind of going against it. Whereas passive transport, you're just rolling down the mountain. There's really no energy involved. And there are going to be 3 different types of passive transport that you need to know. You need to know diffusion, facilitated diffusion, and you need to know about osmosis, okay? So with the diffusion, we're going to just have the simple movement of molecules from high concentration to low concentration. Some things are a little too big and they need like a little help to come in. So we're going to use proteins that are going to be embedded in the cell membrane to let those big things in, and that's called facilitated diffusion. And then we're going to have osmosis, which is going to be the movement of water. Okay, so water likes to go from areas of high concentration to low concentration. So on the next slide, we're going to talk about the 3 different types of tonicities that we have. We have hypertonic, isotonic, and then we're going to have hypotonic. Okay, so I know that this can kind of get a little tricky, but I'm going to try to give you as many like tips and tricks that I can. Okay, so things to remember. Water is going to move from areas of high concentration, or we say water potential, to low water potential. Okay, and I would know these definitions right here. Hypertonic is the side that's going to have the most solute and less water. Hypo will be the opposite, less solute, more water. And iso is equilibrium, things are going to be stayed the same. Okay, water will always go from hypo to hyper. Always, in every situation that you can, because it's going to go from high concentration to low concentration, okay? So if we're going to put a red blood cell, say, into a hypotonic situation, we're going to have a bunch of water like rushing in, and then the cell is going to essentially swell and it could explode. But if it's a plant cells want that. We call it turgid. Our cells like to be in an isotonic state where we kind of have equal movement going in and out, and that's kind of a misconception. A lot of students think that in an isotonic state, you have no movement, but it's no net movement. So you have one molecule moving out and one kind of moving in. Hypertonic is when you're going to have water essentially like stripping and the water is leaving. Our cells are not going to like that. And obviously the plant cells are also not going to like that. But if you notice with the plant cell, they have a cell wall. So everything kind of just pulls away from the cell wall because the cell wall keeps its structure. And that's going through plasmolysis. All right, so let's go ahead and try a concept check. So these are these famous like YouTube experiments, and essentially you have solutes and then you're going to have solvents, and you have to kind of predict which way the water is going to move, okay? And so what you want to do between the two sides is you find the side that has the less solute and then the more water, and then it's going to kind of move into that direction, okay? So if we look at this particular situation, we have pure solvent on the left. So this is all water, okay? And over here is a little bit of water. And so we want to kind of predict which way it's going to move. Beautiful. All right, if we could go ahead and show everybody the right answer that they chose, it's going to go from side A to B. Beautiful. Very good, you guys. All right, so next we're going to talk about active transport. There are 3 types you need to know. So active transport means now we're going against what everything wants to do. So it's going to require energy. Endocytosis, exocytosis, and the sodium potassium pump. Which one means movement of something into the cell? Is it endo or exo? Which one means moving into the cell? Endo, beautiful. Endo is going to be bringing things into the cell. Exo is going to be pushing things out of the cell. The sodium potassium pump is something that's going to help with membrane potential. And it really helps kind of create this charge that we can then do something with. And we see a lot of sodium potassium pumps in the neurons in the brain. And so essentially you just have to move around sodium and potassium, and that requires a little energy. Very good. All right, so let's go ahead and try our concept check. All right, I'll read this out loud because this one's a little bit long. Don't freak out, okay? Look for key words. You can do this. During cellular respiration, electron carriers NADH and FADH2 are oxidized, dropping off electrons at the electron transport chain. Oh my goodness. The movement of the electrons through the embedded proteins provides the energy to push hydrogen ions into the intermembrane space. They gave a little context cue. It is moving against the concentration gradient. What type of transport is this if it's moving against it? Beautiful. Good job, you guys. The answer is A. Good job, right? So it's moving against it, therefore it is active transport. And also, if you guys look at the diagram, I see ATP. I see it. ATP energy. Boom. Easy. Okay. Now let's try the next one. So the next concept check is going to say, once the electrochemical gradient is established, hydrogen ions flow back into the matrix through a process called chemiosmosis. So chemiosmosis is when you're going to have hydrogen ions go from high concentration to low concentration. Passive, beautiful, great job, you guys, great job. And the answer is passive. Good job, everybody. All right, so now if Rashi is willing to unshare her screen, I'm going to go ahead and try to share my screen so we can do a couple questions. All right, let's see how we do. All right, let's see if we can do a couple of questions for Unit 2, you guys, and we are halfway there. How exciting! Your little emojis are making me laugh, you guys. I like it. Keep it going. Look at all those hearts. I love it. All right, a few more seconds for everyone to join. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right, here we go. Let's see how it goes. All right, question 1. Which 2 organelles contain DNA that's separate from the DNA of the cell? So they have their own DNA. We talked about this one. Which one would it be? Very good. Mitochondria and chloroplasts. Great job, you guys. We just talked about that one. Wonderful job. All right, question 2. Which subcellular component is responsible for detoxification and lipid synthesis? Ooh, which one's this one? You guys remember this one? You find a lot of it in your liver. The smooth ER, very good. The rough ER is responsible for making proteins that are then transported out of the cell. So rough ER, think it's rough because it has the ribosomes on it. Ribosomes make proteins. And the ribosomes are bound to the ER, so they are bound to leave. Okay, so it makes proteins that leave. So that's why it is the smooth ER. Which subcellular component is found in both prokaryotic and eukaryotic cells? Good job, ribosomes. Great job, you guys. Let's do one more. Which of the following cell structures is responsible for providing structural support to the cell? Good job. Cytoskeleton is the cell's skeleton. Exactly. It helps provide that nice, beautiful support for it. Great job, everybody. You guys are awesome. All right. So we're going to take a little stretch break. So just take a moment. We are halfway there. You guys are doing phenomenal. Okay. We've done units 1 and 2 now. Okay. Take a moment, have a sip of water, have a nice little stretch. All right, we got this, you guys. We're going to move on to unit 3. Take one more little stretch, stretch your hands, whatever you need to do. All right, my loves, here we go. Unit 3 is cellular energetics. It's going to be enzymes, it's going to be photosynthesis and respiration. Lordy Lou, we got this, okay? Usually this is the toughest of the 4 units, but if anybody can do it, you guys can do it, okay? So we're going to go ahead and have Rashi quiz you on your vocab.
Okay, so just like the last few times, I'm going to read out some definitions and I want you guys to type in the chat what word you think they mean. So the first definition is the breakdown of organic molecules producing usable forms of energy such as ATP. So I'm seeing a lot of cellular respiration, but in this case, the word is actually catabolic. So catabolic means the breakdown of organic molecules producing usable forms of energy such as ATP. And notice how he didn't specify how these organic molecules are being broken down. That would have been cellular respiration. Now, the next example, or the next definition, a site that allows molecules to either activate or inhibit enzyme activity. So I'm seeing a mix of active and allosteric site. So in this case, it's allosteric site. And it's different from the active site because active sites are where substrates will bind on an enzyme. But in this case, the allosteric site is a site where the molecules are they're able to activate or turn off the enzyme activity. So there is no substrate involvement here in the allosteric site. And for the final one, this is used in the process of carbon fixation. And it's in the first stage of the Calvin cycle, and it incorporates carbon dioxide into an organic molecule. It sometimes messes up and picks up oxygen instead. Yep, you guys are right. So this is Rubisco.
Great job, you guys. Well done. All right, let's go ahead and go right into the hard stuff. Okay, we'll start off easy and then we'll get to the hard stuff. So starting off with enzymes. We know enzymes are proteins. We know that they're going to be substrate specific. We just from what Rashi just said, she told you about the active site. The active site is where the substrate is going to bind and then it's going to activate the enzyme. And I want you to remember that they're going to be substrate specific. Okay, so a specific shape has to fit perfectly like a puzzle piece into this enzyme. Anything else that kind of tries to come in is not going to activate this enzyme. Okay, so just remember that they're going to be substrate specific. Enzymes can build things up or they can break things down. So when we talk about all the different chemical reactions, when we are adding water, removing water, dehydration, synthesis, and hydrolysis, all those are going to be mediated through different types of enzymes. Okay, enzymes usually end in ASE. So they are going to be able to, like I said, break things down and build things up. And so there are two terms I want you to know, the difference between catabolism and anabolism. So when catabolism is going on, it's breaking something down, or it's a catabolic process. And when we break something down, we're going to be releasing some energy. Anabolic pathways or anabolism is going to be when we're going to build something up and we're going to store energy. Okay, so when we go from ADP to ATP, we're going to be building, storing energy. That's an anabolic pathway. When we're going to be breaking something down, ATP to ADP, we're going to be releasing energy. Okay, so two processes that we usually talk about are photosynthesis and respiration, right? In photosynthesis, we take energy from the sun and we store it in those bonds. And so that's an anabolic pathway. Catabolic would be like respiration, right? We're now going to take the energy that's stored and we're going to go ahead and release it. And so like my little silly way of remembering is going to be like thinking about a breakdancing cat. Like it'd just be cute to have a breakdancing cat. And so you can think about breakdancing. So breaking down is catabolic. And then I think about a builder and her name is Anna. So Anna the builder. So breakdancing cat and then Anna the builder. All right, so enzymes are going to be able to speed up chemical reactions, and they do that by lowering activation energy. Activation energy is the energy required to start a reaction, and you can see from this graph, I'm sure you guys have seen this graph like a million times. Essentially, with the use of an enzyme, we're going to be lowering the activation energy that's going to speed up the actual process. Enzymes can be reused over and over and over and over. And so I also want you to remember that. Okay, so far I want you to know that enzymes are proteins. They are substrate specific. They speed up chemical reactions by lowering that activation energy, and they can break things down or build things up. Now, there are some terms that you need to know. We can't always have an enzyme on because that would be really, really, really wasteful, okay? And so we have different types of inhibition, okay? And so we have talked about an active site already. So the active site is where the substrate's going to bind. But there's also another site that's kind of like on the opposite of the active site called the allosteric site. And so this is called allosteric regulation, where we have a molecule bind to the allosteric site. And so this creates what's called competitive versus non-competitive inhibition. So competitive inhibition is when you have a molecule bind to the active site, and then it physically competes with the substrate trying to come in. Like the substrate's coming in, it's like bling and it bounces off. Okay, it's physically competing. So competitive inhibition is when you have a molecule bind to the active site and competes with the substrate. Non-competitive inhibition is when you have a molecule that binds to the allosteric site, which is the site that's on the other end of the active site. And when that is bound, it changes the shape of the active site. So now when that substrate comes in, it doesn't fit perfectly anymore. Okay, so that's non-competitive inhibition. So again, competitive inhibition, we have something bind to the active site, physically competes with the substrate. Non-competitive means it's going to bind to the allosteric site, changing the shape of the enzyme, and now it just can't bind anymore. Okay, so now we're going to go ahead and we're going to have Madison do a concept check with you and see if you can really pick that out in a particular pathway.
Perfect. So as you see here in concept check 1, there's a diagram, and we're asking you what type of inhibition is being exhibited in the diagram above. And whatever one you choose, please justify your answer. I'll give you guys about 30 seconds to answer this question. Okay, I think I gave about 30 seconds, and the answer is non-competitive. While Ms. Schumann said non-competitive inhibition is where instead of the inhibitor going onto this active site, it actually goes to a different area of the enzyme. However, it actually changes this, the shape of the enzyme and no longer involves that reaction. I'm going to go give it to Ms. Schumann, to her, if she can explain some of the diagram, if possible.
Yeah, Madison did a great job. If you guys look at this, at first you're kind of like, Lordy Lou, what is happening, right? But if you guys kind of follow it on the bottom and you kind of go left to right, we kind of see that we have a substrate, and kind of falling on the green, that goes into an enzyme. We create another substrate that makes another substrate. This is like every single metabolic pathway in the entire world, right? And then eventually you have this end product that binds to the allosteric site, which will change the shape. And you can see it right here. It no longer fits in it. So that's non-competitive. You guys did a phenomenal job. Great job, you guys. I'm very proud of you for that one. All right, so now let's have Madison go over another concept check with you guys.
Perfect. So this next concept check is discussing the emission of light from fireflies. So I'll read it to you really quick. Fireflies emit light when the enzyme luciferase catalyzes a reaction in which a substrate, the luciferin, reacts to form exoluciferin and other products, which is shown below in figure 1, which shows you reactants and therefore the products. In order to determine the optimal temperature for this enzyme, scientists added ATP to a solution containing d-luciferin, luciferase, and other substances needed for the reaction. They then measured the amount of light emitted during the first 3 seconds of the reaction when it was carried out at different temperatures.
Okay,
So,
I want to add something to this. This is very typical, you guys, of every FRQ and multiple choice. They're going to give you like 12 million paragraphs to read, and you're going to be like intimidated. Like, think of all those big words that Madison just said. Like, oh my goodness, right? So here's my little tidbit of information for you. Okay, I am telling you that if you just look at the figure without any reading and take a deep breath, you're going to be able to answer 95% of the question without having to read any of that background information. So you have to get really good at context clues. So like when I look at this, I go, okay, first thing I'm going to do, I'm going to not freak out, and I'm going to go, they're going to use big words, whatever. That doesn't freak me out, okay? Second thing I'm going to do is say, what are they showing me? So if I look at the figure, look at the caption. If it was a chart, I would say, look at the title. And it says, light is emitted as a result of the reaction catalyzed by an enzyme. And that's what I would read. I can't even say luciferase 10,000 times, right? ACE, we know it's an enzyme. So we literally going to say, light is going to be emitted as a result of this reaction happening. And if you look, you're going to read this left to right. This thing binds to oxygen and ATP, and the enzyme is going to go ahead and do something with it, and we're going to produce light. And that's exactly how I'm going to go ahead and read it, okay? So don't freak out when you guys see big words, long paragraphs. I want you to look at the diagram and say, what are they giving me? And just answer the question, okay? So let's see if you can answer. And then Madison will go ahead and lead you through it.
Perfect. So I'll give you guys about a minute just to answer these two questions, and we'll go from there.
All right, so if you guys look, the rubric is on the screen, okay? So hopefully you can see essentially that the first one is just going to talk about an enzyme. That's it. It just wanted to know the structure. It didn't care anything else except the basic structure of the enzyme. And then the second question said, identify the dependent variable. Well, it talks about emitting light. So they're measuring light. That was it. Nice and easy, okay? On the FRQ, whenever it says to identify, just say the answer. You do not have to put it in a complete sentence, okay? All right, so Madison's going to go ahead and read to you the next two because it looks like some of you guys says essentially that's a little blurry. So if Madison, if you could read C and D to them.
Perfect. Before I go to the next two questions, I just want to let you know, if you guys are confused with what independent versus dependent variables are, independent variables are the variables that are not being changed throughout the experiment, lab, whatever observation you're taking. And dependent variables are variables that are being measured and affected over time, just so no one's confused about those two terms. Great. So the next two questions for concept 2 is C is going to ask you to state the null hypothesis for the experiment, and D is saying that a student claims that as temperature increases, there will be an increase in the amount of light given off by the reaction in the first 3 seconds. Support the student's claim. I will give you guys 30 seconds and I will give you the answers for both questions. Perfect. So I will go now to answering the questions. So C would be that the temperature has no effect on the amount of light emitted. And you guys may be wondering, what is a null hypothesis? Well, a null hypothesis is a statistical hypothesis that proposes that there is not going to be any statistical difference. So there will be no change between the variables. So when you get asked, what is a null hypothesis, you'll be expected to kind of answer in a way as you're kind of not discussing any effects or any results to be made that are significant. Then for D, which is the student claims that the temperature increases and support the student's claim of that. It shows that higher temperatures increases the frequency of collisions and interactions between the molecules, resulting in an increase in reaction rate, meaning that as temperature rises, there will be more kinetic energy between the molecules and therefore the increased rate. And the second answer states that the higher temperature results in a change to the active site that enhances substrate binding. Additionally, when there's high amount of temperature, there will be more enhancement on the substrate binding, which is where the connection between the substrate and the active site is being made, and it kind of shows that the enzyme can possibly denature due to the high temperatures that are being proposed. And now I'll give it off to you, to Ms. Shoemake.
All right, great job, Madison. Thank you. All right. So I'm glad you guys were able to go through for your first FRQ. And what I wanted you to realize is if we didn't read any of that, we were still able to answer all the questions. Okay. So maybe my method of madness is not so crazy. This idea that we just wasted 3 minutes reading these crazy big words when we answered all of them from the diagram. I'm not saying that that's going to be able to be what you can do for every single one of them, but I want you guys to recognize that every minute of this AP exam is very detrimental, okay? So you don't want to read these like huge paragraphs and then get lost in the big words, because I'm an adult and I am a scientist, and when I read these big words, even my head has to process it down. I've now wasted a couple minutes. So my tip for you is when you guys get an FRQ and a multiple choice, if it has a diagram or an image, I want you to look at that first, okay? You go right into that image or diagram, ignore everything else, you take a deep breath, you read, there's always a caption or there's always a title, and you say, what are we breaking down? What are we actually looking at? And I would look at this and go, okay, this binds with this, and here's this enzyme that pushes it all together, and I get this stuff, and it tells me it emits light. Cool, great. Who cares that I don't know what luciferin and oxo? I don't care what that is, whatever. They're just different molecules, but I can see that it produces light. So I'm able, it says describe a characteristic of the enzyme. Ignore the fact that the name is in there. Describe a characteristic of an enzyme. Enzymes are substrate specific. Done, okay? Identify the dependent variable. Well, they're measuring light, so the amount of light. Done. State the null hypothesis. I know the null hypothesis always says that there's no difference. The variable makes no difference. So we would say it makes no difference, okay? And then we are going to go ahead and just support a claim. So I just want you to remember those things when doing your FRQs, okay? Don't freak out. They're going to give you the biggest words possible. So just break it down. Make it super simple for yourself. So continuing our discussion with enzymes, I want you to know that enzymes are going to have very specific conditions in which they prefer, okay? So we kind of all talked about, you know, like enzymes liking specific temperatures and pHs and salinities. And so they are very specific. And when you take an enzyme out of its happy zone, I'm calling it the happy zone, but it's called the optimal zone, then the enzyme's not going to be as efficient. It's going to start to break down, and the breaking down of an enzyme is called denaturation, okay? Yes, our enzymes can go ahead and refold back up if it's not too denatured, essentially. And so let's look at a couple of variables. Here's the effect of temperature on enzyme activity. So if you're looking at this graph, you can see that there is optimal conditions, right at the peak right here. That is the temperature that it prefers. But what I want you to remember, because a lot of students have this misconception, is that they'll always say the higher the temperature and the enzyme's going to denature because it's just going to break down. But think about those enzymes that are in like hot springs. They kind of like higher temperatures, right? And some enzymes like colder temperatures. So instead of always saying that the hotter it gets, it's going to break down, I want you to use the words that enzymes have optimal conditions. And when they are removed from the optimal condition, they're going to start to denature and break down. So when we say denature, that means that those bonds that keep our protein together start to just break. And when you start breaking those bonds, then they're changing the structure. And we know when you change the structure, you then change the whole function of the enzyme. It's no longer going to be efficient until we reach that optimal condition. Think about like when you have a temperature and you're like not feeling good, like you're like, oh, I'm sick. Essentially, that high temperature starts to denature those enzymes in your body, and your digestion doesn't work as well. You don't feel as great, like nothing is working properly. It's not until you get your temperature under control that you're like, okay, I'm feeling better. We have all of our enzymes back into their optimal conditions, okay? So the effect of temperature is going to give you this little bell curve, and so I just want you to recognize essentially what that means and what that is. Same thing with pH. pH is very similar to our temperature. There's going to be optimal conditions. We know that some of our enzymes are going to like lower pHs, which is going to be more of an acidic environment. Some are going to like more basic, right? The enzymes in your, mouth versus in your gut are going to be much different, and you can't put them into the different places. Another thing to think about is the effect of concentration. So if you're asked an FRQ question about an environmental factor that can influence enzymatic activity, your go-tos are going to be like temperature and pH, okay? But if it asks you for a non-environmental factor, then you're going to go really with concentration. That's going to be kind of your key term, okay? So I want you to think about like if 5 of your friends got together and you had one pizza, and you'd probably eat it all within like a minute because there's 5 of your friends and then the pizza. I want you to think about the people as the enzymes and the pizza being the substrate, okay? Now, if I have 100 friends together and one pizza, it's going to be totally eaten within seconds, And then everything just is going to kind of like level off because there's no other work to be done, right? So we can increase the amount of enzymatic activity up until a point, and then it's like all done, right? And so if we wanted to break something down, then we could add more enzymes. Same thing for substrate concentration, right? If I had your 5 friends and I gave you 1 pizza, you'd be like, boom, done, 5 minutes. But if you had your 5 friends and I gave you 100 pizzas, it would take you guys a really, really, really long time for you to be able to break it down. And it would just literally take forever for you to break it down, okay? So concentration has a huge impact on the rate of enzymatic activities, okay? So let's go ahead and do a concept check. So Madison's going to go ahead and do this next concept check with you.
Great. So the concept check is asking you, if an enzyme in a solution is saturated with substrate, the most effective way to obtain a faster yield of products is to... I'll give you guys a few seconds to answer that question. And so I'm looking at the chat, and yes, you would need to add more enzyme. Adding more enzyme can help bind to access substrate, which can further metabolize which can further metabolize for the yielding of the products.
Very good. All right, let's go ahead and move into everyone's favorite, photosynthesis. At least I know it's my favorite of all the reactions, okay? So we know that photosynthesis and respiration are going to be reactions that we say are going to be coupled, right? So essentially, you're going to have in photosynthesis the storing of the light energy into a chemical bond, and then respiration is going to be the release of it. And those two reactions are going to be connected. So we say that they're going to be coupled, okay? And so let's go right into photosynthesis. So for photosynthesis, I want you to know that it's going to store energy. So it's an anabolic process. And it requires light to happen. So we say it's a non-spontaneous reaction. That's just kind of like another term that we throw in there. Non-spontaneous means it requires energy from the outside for it to start, okay? And so in terms of photosynthesis, you kind of have to understand this idea that we're going to be able to do these in those organisms that contain chloroplasts or are autotrophs, okay? So our autotrophs are going to take the energy from the sun, store it into bonds, and then as a heterotroph, we're going to release that energy and use that for something that we can actually do with. So here's kind of the overall process of photosynthesis. It is going to be 2 steps. We have the light-dependent reactions, and then we have the Calvin cycle, which is also called the light-independent reaction, which is also called the dark reactions. Oh my goodness, they all mean the exact same thing, okay? So this first step requires light. I want you to know that the first step is going to be within these thylakoid membranes. And so the thylakoids are these little stacks that you see within the chloroplast itself. And a stack of thylakoids is called a granum. And I think about things like, you know, my grandma makes me a stack of pancakes. So the granum are a stack of the thylakoids. And the process is going to occur all along those membranes. Then we're going to have some products that are then going to be used to drive and it's going to be pushed into the stroma, which is the fluid inside of the chloroplast. And that's where the second step is going to happen, which is the Calvin cycle. Okay. So let's go ahead and go through these processes. And I'm going to try to go at a pace that everyone's okay with. And if you need me to slow down, then just let me know, okay? So let's talk about step 1 at step 1 first. And I kind of get like really excited about this. So like, don't judge me, okay? All right, so step 1, light dependent. So you can see right here, it looks like a phospholipid bilayer because it is, right? So it is going to be the thylakoid membrane. And all of the light is going to be essentially captured by what are called photosystems. Photosystems is a reaction area that is going to have all the pigments. Pigments are molecules that are going to absorb different wavelengths of light. And we have different types of pigments that prefer specific wavelengths of light. We organize them into the reaction center, okay? In the reaction center yourself, we're going to have enzymes. We're going to have special chlorophyll A molecules that are going to absorb this wavelength of light. And then we're going to kind of just absorb it. And so here's the whole process. And it can get a little intimidating and a bit scary, but we're going to break it down just step by step. Okay, so let's go ahead and let's go through each of the processes. So again, this is just step one. Here's like the fancy diagram that you guys might be used to, but I have like broken it down step by step. And so I'm going to kind of just go ahead and tell you my way of remembering it. So if you have a blank sheet of paper and you want to do this with us, it will be a way of remembering the entire process. But if you're someone that just kind of needs to focus along, then just focus along with us. And you're going to want to kind of do it quickly because we don't really have the time for me to wait for you to draw all of it. But again, you have access to these slides, okay? So I want you to think about the first part of photosynthesis like a story. And your job is to remember the story word for word, okay? Because once you remember the story that I tell you word for word, you essentially could draw it really quickly. And then any question I ask you about photosynthesis, you'll be able to tell from your drawing itself. Okay, so here is the setting of my story. Okay, I'm like setting the scene for you. I am going to be at photosystem 2. Okay, there's 2 photosystems and PS2 is actually first. This photosystem is where I was talking about that has the reaction center. It's kind of where we're going to organize all of our pigments in it. And PS2 is first. I know that's confusing, okay? PS2. Here's my thylakoid membrane. And then PS2 is going to contain 2 special chlorophyll molecules called P68, okay? It just means that their favorite wavelength of light is at 680 nanometers. So here's PS2. We have a reaction center that has our primary electron acceptor, and it has P680. That's what we call them, P680. So let's talk about what's going to happen. Okay, so here's the story. Remember the story. Let's look at number 1, and then I'll go to step 2 and 3. Okay, so photons of light excite the pigments in PS2. The energy gets transferred to P680. P680 gets excited, and when he gets excited, he jumps up. And when he jumps up, the primary electron acceptor steals 2 electrons. So it's kind of like a little kid who gets a balloon and they're like, oh, I'm so excited. And then somebody else comes and like pops it. He's mad, right? Like he's not excited anymore. P680 is like, I'm done. Like I'm not doing this anymore. So we have to replace his stolen electrons. So what we do inside of the thylakoid is we're going to have some enzymes that are going to help us split water. So when we split water in that process, we replace the 2 electrons and a proton kind of gets dumped in. And the oxygen that's left just kind of hangs out until it bumps into another oxygen and then diffuses. And that's going to just be what we need to survive. So the oxygen that we get from plants, that's not even the main point of photosynthesis. That's nothing but a byproduct. Thank goodness that's the byproduct, right? Okay, so let's do the first part of the story again. So we're going to go step 1, 2, and 3. Literally follow my arrows. Photons of light excite the pigments in PS2. Energy gets transferred to P680. P680 gets excited and jumps up. 2 electrons are stolen. We have to replace the 2 electrons by splitting water. When we split water, we're going to replace the 2 electrons. We're going to dump in a proton, and then oxygen just kind of gets to diffuse around. All right, so now let's talk about what happens next. And again, yes, I drew these. I'm not the best drawer, but this is just kind of like how I was able to figure it out. So I want you to follow my arrows. All you have to do, look at the diagram, follow my arrows. So let's go to the primary electron acceptor. So find my box in PS2, okay? So the 2 electrons that were stolen by the primary electron acceptor are then going to go to an electron carrier called PQ. Who cares what the long name is? He's just an electron carrier. He does nothing but transport electrons, okay? He's going to take those 2 electrons and drive it through this proton pump called the cytochrome complex. And it does nothing but dump in a whole bunch of protons. That's all that it does, okay? And then PQ is going to pass on those electrons to PC. Who cares that we don't know the names of this? I want you to look at the overall process, okay? So let's talk about the story one more time. Start from the beginning, look at 1. See if you can follow with me. Photons of light excite the pigments in PS2. Energy gets transferred to P680. P680 gets excited and jumps up. 2 electrons are stolen. We replace the electrons by splitting water. When you split water, you get 2 electrons, you dump a proton, and we end up with oxygen as a byproduct. The 2 electrons that are stolen go to PQ. It goes through this pump that pumps in protons, and it goes to PC. So these 3 things right here are literally just transporting those electrons. We call that the electron transport chain. What it does is in the name. Electron transport chain is a chain of different proteins that transport electrons, okay? Now, while all of this is going on, we have another photosystem called PS1. And PS1 is the same thing as PS2, except its special chlorophyll A molecules are called P700 because it likes the 700 nano wavelengths of light, okay? So while this is going on, right, everything's happening, we're going to have photons of light excite the pigments in PS1. All of that is going to get transferred to P700. The same process happens. P700 gets excited. The 2 electrons are now going to move into the prime electron acceptor, okay? Then our P700 is now sad. We have taken his electrons, so we have to replace his electrons. And for this one, right next door to him is PC. PC is just holding the electrons, and so he sees that P700 is upset, and he's like, chill, it's okay, and goes boop, and just hands it over, okay? So I want you to know that that's a difference between PS2 and PS1. In PS2, when P680 got the electrons stolen, we split water. And PS1, the same process is happening, except we're going to replace his electrons by PC. Okay, so now the 2 electrons at the electron acceptor in PS1 are going to go to another carrier called FD. Ignore the names, okay? And FD is going to use those electrons to activate an enzyme called NADP reductase, creating an energy molecule called NADPH, okay? And in that process, it dumped in a proton. So up to this point, we have had protons get dumped in 3 times. And you should know the 3 different times they got dumped in. The splitting of water through that special pump and the creation of NADPH, okay? So now we have a super high concentration of protons on the inside. Like it's super, super duper high. It's like busting at the seams. So what's going to happen is all those protons are going to want to leave. And the only exit we have is an enzyme called ATP synthase, which creates ATP. So all those protons are going to go from an area of high concentration to low concentration. And it's going to diffuse through this enzyme creating ATP, bringing the protons back to the outside so that the whole process can happen again and again and again and again and again, okay? So now, let's see if we can say the whole story again. So let's start from the beginning, and let's see if you can say it while I say it, okay? Here we go. Protons of light excite the pigments in PS2. Energy gets transferred to P680. P680 gets excited and jumps up. 2 electrons are stolen. We replace the electrons by splitting water. When you split water, you replace the electrons, you get a proton, and then you get some oxygen. The 2 electrons go to PQ, they run through a pump and end up at PC, and we dump in more protons. At the same time, you're going to have photons of light excite the pigments in PS1. Energy goes to P700. P700 gets excited and jumps up. 2 electrons are stolen. We replace P700 electrons by PC, right? The dude just goes, boop, there you go. Those 2 electrons then go to FD. We then are going to run it through this enzyme creating NADPH. Now we have a super high concentration of protons that want to leave, and they're going to diffuse through this enzyme creating ATP. This movement of protons is called chemiosmosis. Osmosis is the movement of water, movement of chemicals, right? The movement of those protons are going to create ATP. So what I want you to know is that the 2 products created from the first step of photosynthesis are ATP and NADPH, okay? Those 2 products are needed for the next step of photosynthesis to occur. It just kind of gets dumped into the stroma, which is the cytoplasm of the actual chloroplast itself, okay? Now, for the next step, we actually are going to need more ATP than we do NADPH. So what's going to happen is ATP is produced from the movement of protons. So what we're going to do is we're going to go through what's called the cyclic electron flow. So instead of my 2 electrons going to FD and then activating NADP reductase to create NADPH, that's just a waste. It's going in one direction. Instead, we just go backwards through that cytochrome complex pump, and we just pump in. We go pump, pump, we go over and over and over. It dumps in the protons. Now we have a high concentration. Chemiosmosis wants to happen, and we're going to run right through creating more ATP. Okay, so I need you to know that NADPH essentially is going to be created through this electron transport chain. And ATP is really created through that movement of chemicals, which is called chemiosmosis. Okay, that's step 1, right? That all requires that movement of light. Step 2, I know it gets like a little scary, but like, don't be scared, okay? So what's going to happen for step 2, this is called the light independent reaction. So for this part, it's going to be in the stroma. We no longer directly need light for happen. We just need that ATP and NADPH. This looks a little scary, but I want you to take a deep breath, okay? So the point of this is to create essentially a little baby sugar. And we know that plants are going to create something called glucose, and that's how it's going to store its energy for when it needs to be able to break something down, okay? So a baby sugar is called G3P. Who cares what it stands for? Okay, a G3P is a cute little baby sugar. 2 G3Ps are going to give us 1 glucose, okay? So what happens is you have an enzyme called Ribisco, okay? Ribisco is going to combine 3 CO2 molecules, and it's going to end up, and that's called carbon fixation. So 3 CO2 is going to bind to this enzyme, and we're going to create a molecule that's kind of like big now, and we're going to use a little ATP, a little NADPH, and we're going to just rearrange the molecule, okay? So when we end up rearranging the molecule, we're going to break off a little baby sugar, and then we have to restore our molecule back to a phase where it can now accept more CO2. So we just kind of have this cycle going over and over and over through this enzyme called Ribisco that's going to be able to combine the CO2 to the actual molecule itself to create this little baby sugar. And that's the whole point of this whole thing, okay? And in order to regenerate our original molecule so that it can accept CO2, we have to use a little more energy, because every time we break something down or build something, we know that's going to require energy. Okay, so that's the second part. Okay, so that's photosynthesis. The other thing I want to go over also about Ribisco is that Ribisco is a very, very old enzyme. It's been around for like a really long time. And so what can happen is Ribisco can actually bind to oxygen in addition to CO2. And so we have a structure on the bottom of a leaf that can open and close. And what's supposed to happen is we're supposed to have oxygen leaving, and we're supposed to have CO2 going into it so that the CO2 can bind. And that's why plants need us to talk to them, right? They use the CO2, they pulled out of the atmosphere in order to create the sugar for themselves. And so does anybody remember the little structure under a leaf? It looks like a mouth. What was the structure under a leaf that controls gas exchange? It looks like a little mouth. Does anybody remember? What are the little mouths under leaves called? Yeah, the stomata. Very good. Good job. Yep. Did you guys know that that is one of my favorite all-time favorite science words? So part of my, I'm part Italian and the other part of me is Chinese. And so whenever I say stomata, I have to say it in like a real thick accent. So I always say like, it's the stomata. Like I just sounds like my family. So the stomata is going to be what controls gas exchange. And so if it's a really hot day, the stomata is going to close by the guard cells, and it's going to kind of trap those gases in there. So now oxygen can't leave. Oh no. Okay. So when oxygen can't leave, we have oxygen bouncing around, and they can actually bounce around and combine to Ribisco. And so that is called photorespiration, which is not good. And so plants have to figure out a way to prevent oxygen from binding to ribisco, because ribisco is an old enzyme and he's like, come on in, it's fine. But if oxygen binds, it's going to end up making something that will break down the sugar and can actually kill the plant. Okay, so a couple of things that plants have done to prevent that from happening is they've been able to partially close. So instead of opening all the time, it closes, or they change the time of day, like cacti are going to change the time of day. So those are just different ways to prevent from that photorespiration process from happening. All right, so let's go ahead and let's move on. Guys, I'm having some technical difficulties. Give me 1 second. All right, here we go. So now let's talk about respiration, okay? So this is what we're gonna go through, okay? You guys hold strong. It's almost 10 if you can even believe that, okay? So photosynthesis takes the light energy, stores it into a chemical bond, okay? As a byproduct, we get oxygen, thank goodness. Respiration is going to be now to release all of that stored energy, okay? And respiration is very essential. Respiration is going to occur in the mitochondria. So any organism that contains mitochondria is going to be able to go through cellular respiration, whereas photosynthesis only occurs in our autotrophs, okay? So plants can do photosynthesis and respiration. We can only do respiration. So you have to kind of know the organelles that's involved in it. So in the mitochondria itself, we have different parts of it. And what you'll notice is that the mitochondria is highly folded, just like the chloroplast had lots of thylakoids in it. And what we have done is we've increased surface area. And when you increase surface area by having all these folds, it allows for more chemical reactions to occur. And so the cell is able to be a little more efficient. Okay, so what we're going to do is we're going to go into the different parts of the mitochondria. It's a double membrane structure. And if oxygen is present, that's called aerobic respiration. And we're going to be able to move into the mitochondria and get the most amount of ATP. So I want you to know that there are two types of respiration. There's aerobic and then there is anaerobic respiration. Aerobic respiration requires oxygen. Anaerobic does not. Okay. So when I look at the different steps of respiration, the very first step is called glycolysis. Glycolysis is the first step, whether oxygen is present or not. I want you to know that glycolysis is going to actually happen in the cytoplasm itself. And it's an anaerobic process. And from the whole process of glycolysis, we're going to get 2 ATP. It's not very efficient, but we do get ATP from that process. So if you guys look at the word glycolysis, lysis means to break down, glyco means glucose. So glycolysis is literally breaking down glucose, okay? So we're taking a 6-carbon molecule and we're just gonna kind of rearrange him and break him apart into 2 3-carbon molecules. Every time you take an organic molecule and you change the amount of carbons that are there, you have to give it a new name. And that was the hardest part of organic chemistry. Whew, in college, you guys, wait till you get there. But anytime you change a molecule's amount of carbon, it's going to go ahead and get a new name. If you look at the starting molecule and the end molecule, if you have released a carbon, like you go from 6 carbons to 5 carbons, I want you to know that one of the rules of respiration I want you to remember is that when you go down the amount of carbon, it's released as CO2, okay? But in this process, I have a 6-carbon molecule that I literally chop in half and it becomes 2 3-carbon molecules. So I still have 6 carbons just chopped in half. It became smaller. Every time you break a bond, you're going to release some energy, okay? So that's kind of where that energy comes from breaking that bond. Now, if the oxygen is present, we're going to get pulled into the actual mitochondria itself, okay? So this diagram right here, and it's on your note sheet, is going to show you the 3 parts of cellular respiration, okay? We have glycolysis, the citric acid cycle, which is also the Krebs cycle, and then we're going to have our oxidative phosphorylation, okay? And so this only happens in the presence of oxygen. So we just did step 1, glycolysis. 6 carbon, boom, broken down into 2 3 carbons. We have broke a bond, energy is released, but we have the same amount of carbons. Those 3 carbon molecules are now called pyruvic acids or pyruvates. So there's 2 of them, okay? They're too big to go inside. We want to draw them inside. They're too big of molecules. So we're going to have to go through an enzymatic process. We call it like the transition step or the intermediate step. It means the same thing. Essentially, you're taking a 3 carbon molecule and you're turning it into a 2 carbon molecule. So going from step 1 into step 2, I have to release a carbon for it to fit. It's too big, okay? So now I've released some CO2 into the atmosphere, essentially. And I'm going to go ahead and add something that's needed for my next step, okay? So it's going to be called an acetyl-CoA. It's a coenzyme A. So our molecule went from a 6 carbon molecule called glucose to 2 3 carbon pyruvates. Now there are 2 2 carbon molecules called acetyl-CoAs. They're just different names, okay? So now we've reduced the amount of carbons. That enzyme is then going to be needed to go into the matrix of the mitochondria to go through what's called the citric acid cycle. And in this step, we're going to be breaking down every single thing that's left, okay? We're going to fully break it down. There is no energy left. It's boom, gone, okay? So the rules of respiration I want you to know is, number 1, when you change the amount of carbons, you call it a new name. If you're going to reduce the amount of carbons, it's going to be released as a CO2. And the third rule I want you to remember is every time we break a bond and energy is released, we have what are called electron carriers to capture those electrons, okay? And our 2 electron carriers are called NAD and FAD. And I love that they kind of rhyme, NAD and FAD. Okay, so NAD and FAD are nothing more than capturers of any electrons that are released from a bond. Any energy that's released, we capture it up, okay? So in this first step, when I told you that we went ahead and we went from a 6 carbon molecule to 2 3 carbon pyruvates, no CO2 is released because it's still 6 carbon, but we did break a bond. And so we're going to have NAD there. NAD's going to capture it. And now that he has gained an electron, we're going to call him NADH. He's going to take that and drive it into the inner membrane. When we then go from our 2 3 carbon molecules, we're going to come to that intermediate step. It's going to become 2 2 carbon molecules. We release some CO2. We're also going to have NAD there. NAD's going to capture that and also take that was released. And in this last step where we fully are going to break down everything, there is nothing left. No energy is left in our original glucose. That boy is gone. He's long gone. Okay, poof, gone. Here we have another electron carrier, which is FAD. So FAD's also going to be captured. So it's going to just kind of be like, if you guys imagine like those little machines where you go in, I've never really seen one, and it releases money, you try to capture all the money that's being released and you get to keep it. It's the same thing. In this step, we have a bunch of enzymes chopping up our molecule and releasing everything. There's nothing left. And NAD and FAD are there just capturing everything, okay? So we're going to take all that and we're going to bring it into this inner membrane, into the very last step, which is oxidative phosphorylation. Essentially, we're going to go through the electron transport chain again. We're going to use chemiosmosis again, and we're going to create that proton force to generate ATP. So this is aerobic respiration, glycolysis, citric acid cycle, oxidative phosphorylation. We produce 2 ATP from step 1, 2 ATP from step 2, and the most amount of energy that we get is going to be through step 3, through that proton gradient, okay? Using the movement of protons to create the most amount of ATP. And again, this happens only in the presence of oxygen. Now, let's say oxygen's not available, okay? In that situation, that is going to be called anaerobic respiration. So oxygen's no longer available, okay? So in this situation, we don't get pulled into the actual mitochondria. We have to kind of stay on the outside. So imagine like you're running and all of a sudden you like feel like you hit a wall and you're grasping for air. You're like, oh my God, I can't breathe. Some of us, that happens in like 10 minutes and some of us happens like in hours. I'm not a very good runner. Sure, you guys are like way better runners than I am. But at some point you start grasping for air. At that point, you're in anaerobic respiration. You don't have enough oxygen moving those electrons into the mitochondria to generate that max amount of ATP. But you're not going to keel over because your body will tell you to stop. So then you stop, okay? So I just see somebody ask me how many ATP is produced in the last one. It's 32 to 34 ATP gets produce. So it's 2, 2, 32 to 34, okay? So in this step, what we're going to do is we're going to go through what's called fermentation. We have lactic acid fermentation, and then we're going to have alcoholic fermentation. So again, oxygen's not present, okay? We still go through glycolysis. That's the same process for aerobic and anaerobic. The difference is we're going to go through a series of enzymatic reactions that are going to regenerate NAD, so then he can then go back and continue to accept more and more and more electrons. So the point of anaerobic respiration is to go through glycolysis over and over, but you only produce 2 ATP at the same time. We go through what's called lactic acid fermentation. When we go through this process, one of the products is called lactic acid. And the lactic acid is kind of like when you feel the burn essentially in your muscles, even though it's also micro tears. Yeast and bacteria are going to go through alcoholic fermentation. One of their products of this process is going to be alcohol and CO2. So they actually go through like a reduction of the actual carbon itself. All right. So let's go ahead and let's have Madison do another concept check with you guys.
Perfect. So I'm going to give you 3 minutes to answer this concept question, and it's asking you to use each of the following observations to justify the claim that glycolysis first occurred in a common ancestor of all living things. So you see the 3 observations below, and you have to justify the claim. I'll give you guys, once again, 3 minutes. So my time is currently 8 o'clock. So whatever your main time is at 31. We'll get back to you guys, and I'll go over the answers.
All right, we are at 9:30. So Madison, can you kind of go over where they would have gotten points for this question?
Yeah, of course. So for the first observation, it discusses on more the inheritance of the gene trait process and its origination of it. And the second justification will discuss more of like the selective advantages you can have with glycolysis. And the second observation then kind of discusses further on the origin of glycolysis, the predates to like the free atmospheric oxygen / photosynthesis, since anaerobic conditions mean that there's like no that oxygen has not appeared more within like the production photosynthesis. So in simple terms, there's no oxygen, what it means for anaerobic. And lastly, for the last observation, where it discusses cytosol, which is the type of fluid in cytoplasm. It will discuss once again the origin of glycolysis, predating to the cell types with the memory-bound organelles.
Very good. Great job. All right. We are going to take a mini stretch break. You guys, we have 1 unit left. Have a nice little stretch. That was the hardest one. I know. Okay. We didn't have time to do the practice quizzes, but once you guys go into your Fiveable and you click on AP Bio, you can take all these questions. All the questions are there for you. It's the same questions that I pulled from. Okay. All right, so let's take a little break. You guys are doing an amazing job. I'm very proud of you, okay? You got this. So we got this. Chemistry of life, macromolecules. We talked about cells, and then holy moly, cellular energetics. Luckily, you guys, this is the easiest part. Thank goodness. And this will be the quickest part, okay? So we're going to go ahead and go right into cell communication, the very last part. You can totally do this, which is unit 4. So for unit 4, it's kind of a smaller unit, and a lot of times your teachers usually do this in like 2 days because it's kind of like really easy. And so here are the key vocab. So I'm going to go ahead and ask you which one of these words, let's see, let me pull it up. is going to be the first stage in mitosis. What is the first step or stage of mitosis? Does anybody remember? Prophase, prophase. Good job, you guys. Great job. Okay, so prophase. What's the next step? What happens after prophase? What happens after prophase, you guys? You got this. Metaphase. Beautiful, beautiful, beautiful. Okay, what happens after metaphase? I'm bringing it back to easy questions. What happens after metaphase? Yes, after prometaphase, I should say. Anaphase, very good. What happens after anaphase? Yes, very good. Telophase. Okay. Great job. You guys are rock stars. Okay. Easy peasy. All right. So now let's go ahead and let's talk about how cells communicate. Okay. There are 4 different types of communication that we can have. We can have endocrine, paracrine, autocrine, and juxtacrine. Endocrine communication is long-term communication, okay? So that's essentially like if your body is to release a hormone that's going to travel through all the cells over your body, it's going to travel very long distances, and only the cells that have the receptors that are specific for that hormone, and whatever is binding to a receptor we call a ligand, which is a fancy name of saying the chemical that's binding to the receptor. That's called endocrine. Paracrine is where we're going to have some local communication. Autocrine is going to be some self-communication. It's going to tell itself to grow, to not grow. And juxtacrine is when we have some kind of that contact. So like in your heart cells, you're going to have your cells that are going to talk to each other. Or in plants, you're going to have that plasmodesmatas, which is the little channels between plant cells so that they communicate with each other. So 2 keywords I want you to know is a receptor and a ligand. The ligand is nothing more than what's going to start this whole reaction to happen. It's a chemical that binds. The receptors are going to be what the ligand is going to bind to. So when we receive information from the outside and we need something to happen on the inside, that's called a signal transduction pathway. And it's going to occur in 3 steps. We have reception. We're going to receive that information. Signal transduction is nothing more than a series of enzymes activating other enzymes to essentially turn other things on. And then we're going to get some sort of response that's going to happen, some sort of response, okay? So my idea is like if someone was to ring the doorbell, you're going to hear the signal, and then someone's going to come to the door, and maybe it's like your best friend that's like, hey, can you get so-and-so? And then your mom's like, hey, so-and-so's here. That amplification, that's sending that signal is the transduction. And then there's going to be a response. Like you're either going to be like, I'll be down in a minute, or like tell them to go away, however you're feeling. You know what I mean? So that same thing happens in the cell. So we're trying to get information from the outside of the cell to inside of the nucleus so that something can actually happen. Okay. And so that is going to be your signal transduction pathway. How do we get our cells to communicate from external things to the actual inside itself? Okay. So cells are going to communicate with themselves, and they're going to communicate with others as well. All right. So let's go ahead and let's talk about what's called feedback. Okay, so feedback happens within organisms that want to maintain homeostasis, right? So sometimes we're going to have to respond to things in a certain way. Sometimes it's going to be amplification will be how we're going to respond. And so what I want you to know is the difference between a negative feedback loop and then a positive feedback loop, okay? So a positive feedback loop is amplification. When we receive information from the outside that we need to continue to do something in one direction, that's considered positive feedback, okay? Negative feedback means we're receiving information from the outside and we've kind of gone beyond a point and we have to come back to it. So it's kind of like, ooh, we fluctuate. We kind of go up and down, okay? And so we receive the information and the response will be go up and down. That's going to be negative feedback, and positive means just keep going. So my 2 examples of like a negative feedback loop is going to be something essentially like body temperature, right? When you get too hot, your body's like, oh my gosh, I have to find ways to cool down. So you essentially have a set body temperature, and when you go too hot, we respond by essentially sweating, right? If we get too cold, we respond by shivering. But when we come to that point, that's called negative feedback. Positive feedback is just full-on amplification, okay? So the example is going to be like the ripening of fruit, right? So you've never had a banana go from green to brown and then go back, because that would never happen. But that one movement beyond a set point is called positive feedback. So those are kind of different ways that we can respond to communication from outside. All right, so now I want to talk about the cell cycle, okay? So cells, we know, grow, and they have to be a certain size. So cells can either divide or they're going to die. So cells are going to go through what's called the cell cycle. And most of the time, your cell is just growing in what's called interphase, okay? Does anybody remember the 3 parts of interphase? There were 3 parts of interphase. It's also in the diagram. What are the 3 parts of interphase? Beautiful. Great job, Amy. Great job, you guys. G1, S, and G2. Beautiful. Okay. G1 means the first time it's going to grow. S means DNA is going to be synthesized. G2 means the second time it's going to grow. And each time we go between those steps, we have certain checkpoints, right? So we have a cell that's going to grow, and then we're going to have to kind of make sure that it's growing like it's supposed to. And then it can move on to the next step and the next step. Once we go through interphase, once we get big enough, we now need to divide, and that's called mitosis, okay? So once we grow, it's interphase. Then we go through mitosis, which is prophase, metaphase, interphase, and telophase. What is the final step, which is the division of the cytoplasm? What is called the division of the cytoplasm? What is the very last step of the cell cycle? Does anybody remember? Cytokinesis. Beautiful. That is the division of the cytoplasm. Great job, you guys. Beautiful. All right. Now let's go into the actual phases. Most of the time, you guys are great with this. You can identify the different stages, which is nice. But what you usually forget is the terminology. Okay, so here's some terms that I just want you to know the difference between. So usually if I asked you to draw a chromosome, you would draw the letter X. And in between, you put like a little dot. Okay, so what holds these 2 individual arms together is going to be called a centromere. The centromere is in the center. And each individual arm is called a chromatid. And when you have 2 of them together, they're called sister chromatids. So this arm matches this arm, and it's held together by this little center part right there. Okay. So what I want you to know the difference between is the chromatin and the chromatid. Chromatid is coiled up DNA. Chromatin is uncoiled DNA. So most of the time when your cell is just like chilling, your DNA is just kind of like a bowl of pasta. It's chromatin. But when it is time to divide, you want to organize all of genetic material into some sort of easy package that can easily be transported. And so that's when it coils up into a chromatid. So you can't actually see the chromosomes until you have mitosis begin. Up until that, it just looks like a big bowl of pasta, like I said. Okay, so make sure you know the difference between those terms. Now, we are going to go ahead and try some practice questions. Okay, so let's see if we can do some practice questions. So give me 1 second. Take a little stretch if you need it. We're on the home stretch. We got 20 minutes, you guys. We totally got this. All right, so let's go ahead and see if we can answer some of these questions. Here we go, y'all. Last part. We can do it. All right, you guys, hold strong. I am so proud of you. You are a rock star. We have gone through units 1, 2, 3, and 4. Here we go. Hold strong, you guys. I know you're tired. It's been a long week. You and me both. Trust me, I get it, y'all. Thank you for those that woke up at like 2 a.m. I know that was so hard. I appreciate you. All right, let's go with 10 more seconds and then we'll go ahead and start the quiz. I'm not seeing any more hearts. Where are you guys at? I'm seeing a lot of chili peppers. Does that mean you guys are tired? There the hearts are. Thank you guys. Thank you for sending me some love. All right, my love bugs. Here we go. We got this. All right. Which type of cell signaling is always sent through the bloodstream through long destinations? So which one travels long distances? Which one's long distance communication? All right, let's see. Endocrine. Great job, you guys. Well done. All right, let's try the next question. What is cell signaling and how is it important to the development of organisms? Ooh, this is a tricky one. It's taking you guys a while. See? Beautiful. Great job, you guys. Well done. Let's do a few more. Another name for a cell signal or stimulus molecule. What do we call that molecule that binds to the receptor? What was it called? A ligand. Good job, you guys. In terms of these other terms, I just want to make sure you kind of understand what it means. A receptor protein, just look at the word. It's a protein that's going to just receive the ligand, okay? Kinase essentially means where you're going to have a series of enzymes kind of activating the other enzymes. Phosphorylation cascade, ooh, that's a big word. Basically, it's very similar to the kinase. It's almost the same thing, where you're going to have one molecule phosphorylate another, which activates another and activates another. All right, let's try one more question. A cell sends a chemical message that targets itself. What type of signaling is that called? What's it called when it targets itself? Autocrine. Great job, you guys. You did a phenomenal job. All right. We are now going to go ahead and I'm going to have Rashi kind of give you some final words. She's going to go ahead and just kind of give you some words of advice. And then we're going to kind of end with the last thing that we're going to do will be pure questions from you guys. And we'll be there to help you guys with that. Okay. So Rashi, they're all yours. Let me pull up your slides so that you have them. I'm getting there, Rashi. Apologize. All right, Rashi, my dear, the slides are not wanting to work, so they are all yours. I'll work on it.
That's fine. I'll just talk then. So first of all, I am super proud of you guys for making it through these last 3 hours. I know that it is difficult to sit here and live stream about a topic that you're not very super passionate about or you're just really stressed about, but you guys managed to do it. Some of you guys are up at like your 2 a.m., which is crazy and insane. So super proud of you guys for managing to make it this far. Now, some great resources. So a lot of you guys have been asking for more resources in the chat. Number one, you are on a Fiveable. So of course, I'm going to recommend the Fiveable study guides. They have study guides for nearly every unit in AP Bio. So if you don't understand a specific topic, like maybe you don't understand what a double bond is, there's a study guide that addresses exactly that with a lot more practice problems. Plus, there are other videos and other information that expands on these topics. So make sure to go check those out. And if you're someone who prefers maybe more of like a physical textbook, the Barron's and Princeton textbooks are really great resources to help you study for this. And don't forget about your free AP Classroom questions. AP Classroom, I know maybe you're not the biggest fan, but it is incredibly useful. It has lots of important questions and it goes over those questions with the answers really nicely. The best part is those questions are written directly from College Board, so they're a very accurate representation of the types of questions you might see on the AP exam. So I highly recommend you go and check those out. Now, there are a few more crams coming up in the next few days, so make sure you guys are here again in 24 hours to go over units 5 and 8 tomorrow. And then on, Sunday? On Sunday, there will be Skills and Drills Part 1. On Monday, Skills and Drills Part 2. And then next Tuesday is the grand finale. So your AP exam is in 5 days. And what does that mean? That means that you have 5 days to cram with us. Here at Fiveable, we fully believe in you guys. The fact that you guys are here giving up your Friday nights or your Saturday mornings or whatever time you're in to study for AP Bio tells us how much you guys are willing to work hard. And so I fully believe in you guys, and everyone else here fully believes in you and your capability to absolutely crush this exam. So don't freak out, because we're here to help. There's a lot more people in the chat who are talking about seeing you guys in 21 hours. You're not alone, and we have your back. And lastly, make sure you follow @ThinkFiveable on social media for more practice like this. Plus, I know a lot of you guys are doing a lot of other different AP exams. Fiveable has some great study guides and review material for those as well. So go take a look at that. Fully believe in you guys. You guys are going to do great. And thank you all so much for joining today.
Thank you so much, you guys. We are so proud of you. Feel free, we're going to stay on for a little bit to go ahead and ask some questions in the chat, and then we're all going to be able to answer them. And for those of you that want to do a little more practice problems, I'm going to go ahead and just share my screen for like another quiz if you wanted to join the quiz, and just I added all the questions and see if you want to go ahead and try the questions. So we'll leave it up to you. You can either say, oh my goodness, I did it, and take a nap. You can also ask questions in the chat, and then I'm going to go ahead and share my screen for anyone that wants to do a last 10-minute review of all the questions, and just to see how you would be able to do if I mixed up all the different ones together. Okay, so let me go ahead and see if I can pull it up for you. Take a deep breath. You guys you did so good. I'm so proud of you. All right, so here is the final review for anyone that's like, let me do this. I got 10 minutes left in me. I can totally rock out with this. Go ahead and join if you would like to do the final review with us. Let's just finish strong. It's okay if you need to take your break and call it done. I look forward to seeing you at our future crams. Everyone else, we're going to hold strong for 10 more minutes, okay? So I have put all the questions together. There are those hearts, you guys. Hold strong. I'm very proud of you. Thank you for the love. If I could send hearts back, I would send hearts back to you guys. All right, let's see how many we can do. Okay, we're just going to keep on going until 10. So good luck, you guys. All right, here we go. All right, 2 students are arguing about which elements are most prevalent in living things. Which element symbols best summarize the elements found in living things? So which one of those, A, B, C, D, are the main elements found in living things? It's, oh, you guys, look at that, a little tie. So it is chinops. That's just kind of how you remember it. Those are the main elements, but I could see how that's tricky because carbon, hydrogen, oxygen are also the main elements. So I'm going to say that you guys are all right. So good job, you guys. You rocked it. All right, second question. Which statement best explains why water molecules can stick to other surfaces? So adhesion. All right, very good. All right, so C and D. So D says, as a polar molecule, water is attracted to other polar molecules. So remember, it's about the charges, and that's what is going to allow the molecules to stick together. Remember, it sticks together through the hydrogen bonds. And so I think that's where the confusion was, because this one said covalently, but it's really like the hydrogen bonds. That's what's in between two of them. That was kind of tricky. All right. Although hydrogen bond results from polarity of oxygen and hydrogen atoms in water molecules, the molecule itself is held together by what? So the bonds between the hydrogen and the oxygen within the water itself. What type of bonds is that? Please don't tell me London. Do not say C. That's made up. I don't even know what that is. Okay, so the hydrogen, oxygen within the water molecule itself. We know that hydrogen bonds are between. What about within it itself? It's a very strong bond. Beautiful covalent bond. Thank you for not choosing C. London forces, Lordy Lou. All right, here we go. Some insects can walk on water without breaking the surface. What property of water is responsible for this? Oh, you guys answered this one so fast. Good job, you guys. Surface tension, and you all got it right. Good job, everybody. Great job. Let's try the next question. Polarity, a property of water, is caused by the unequal distribution of charge between oxygen and hydrogen. While hydrogens are positive, oxygen is highly negative due to the elements blank. Good job. Electronegativity. Good job, good job. Those are my chemistry people. Great job. Lipids may be straight or branched. Saturated lipids, sat fats, are considered less healthy and consist of what type of bonds? So sat fats have what type of bonds? Remember SSS. They're solid at room temperature because they contain these type of bonds. Good job, right? So only single. So SSS, saturated fats are solid room temperature and contain only single bonds. Great job. All right, next one. What is the level of protein that allows multiple proteins to bind together? We did this one. Let's see how well your memory is from like 3 hours ago. So it's multiple polypeptide chains coming together. Was that primary, secondary, tertiary, quaternary? Yay! Good job, you guys. Quaternary, great job. Primary is a linear sequence. Secondary is when it kind of has local folding. Tertiary, the R groups interact, and quaternary is when you have multiple proteins coming together. You guys are doing so good. Hold strong, okay? 3 more minutes. Let's see. Amino acids string together to form the primary structure of proteins. What type of bond holds together amino acids? What was the name of that bond? Peptide. Good job, you guys. Great job. All right, next question. Let's see what we got. Which type of biological macromolecule is composed of long chains of amino acids? You better know this one. Which one of those macromolecules contain amino acids? I better see that answer quick. A bunch of amino acids make a protein. A protein. All right, here we go. Which of the following is a characteristic of carbohydrates? So carbs. I think pasta, which I love pasta. The Italian side of me. A source of energy. Great job. Hydrophobic is lipids. Insoluble in water means the same thing as hydrophobic. So those are lipids. And then we just remembered that amino acids make our proteins. What is the primary function of proteins? This will be the last one we do together. Enzymes. Very good. I can also see membrane structure. So if you chose A, I'm going to say for sure. Information storage, we know, is going to be our DNA and RNA. Energy, we know, is going to be our lipids. But so really it could have been A, B, or D. So if you guys chose any of those, then you guys are rock stars. All right, my loves, it is that time. Okay, I am proud of you. I hope you will continue to be in the Crams with us, and I hope you guys have a fabulous